Okay, everyone, it's time for us to start. So thanks for joining, and it's great to see so many familiar faces and some new faces as well. So that's really great. So welcome all to the 2024 Dundee Root Medal Lecture and Workshop. We'll begin with the main event of the day today, which is a lecture from Amelia Henry, who will present her lecture, which is Integrated Root Phenotypes Under Seedling Stage Drought in Direct Seeded Rice. After which, we'll ask her to take the floor and receive the Dundee Root Medal. Once the formalities are over, we'll keep you interested with some talks from friends and colleagues of Amelia. And then after lunch, the lunch break, at least for lunch in the UK, we'll hear from some of our colleagues working in Scotland on our favourite subject of roots. The main event of the day, which is the award of the Dundee Root Medal to Amelia Henry. Amelia is a senior scientist in stress physiology at the International Rice Research Institute in the Philippines. She's the unit leader for traits for challenging environments, and her group conducts physiology experiments on rice, which aims to identify new accessions for stress tolerance and to characterize variation in the breeding pool. Research topics include drought, salinity, heat, direct seeding, lodging, and rationing, with an emphasis on characterizing rice roots. Amelia works closely with national partners to characterize stress-prone environments and build capacity for stress screening. The aim of her unit is to link upstream research with breeding through the trait development pipeline. So let's welcome Amelia to share her slides and present her Dundee Root Medal lecture. So over to Amelia.
Okay, well, thank you so much, Tim, for that nice introduction. I'm very pleased to be giving this presentation today, and I would like to thank the Dundee Root Medal Committee of the International Society of Root Research for this honor. The topic of my talk is integrated root phenotypes under seedling stage drought in direct seeded rice. And to start with, I'd like to give a general background about where I'm coming from with this work. So I work at the International Rice Research Institute, which is headquartered in the Philippines. Our aim is to alleviate poverty and hunger through rice science. And you can see we have a more detailed description of all the different aims that we have now. So many people may have heard of IRRI for our work in the 1960s related to the Green Revolution, for our gene bank, which holds many diverse traditional varieties, or for some of the management strategies that we have developed, particularly alternate wetting and drying. But maybe not everyone is aware of how IRRI has a long legacy of root research. So here's some photos from some of our older annual reports, as well as from some colleagues that have already retired. You can see some of the work that was done back in the 70s and 80s. We have some images of root pulling force that was used in the field to estimate root architecture in the context of drought stress. There was also a quite elaborate aeroponic setup back in the 70s. And both of those strategies were used to screen hundreds and hundreds of diverse rice genotypes. And here in this photo on the right, you can see a monolith. So that was a form of root sampling that was used in the field with the idea that by having a square around an individual plant, we would get a representation of an entire root zone. So that's some of the more whole plant field-based work that was done here at IRRI in the past. But we also have quite a history of more molecular and genetic studies related to roots. So here's some examples, including inducing rhizobial colonization in rice, work on phosphorus deficiency, genes related to drought tolerance, and genes and QTLs related to direct seeded rice. All of this work was focused on roots and also involved transgenic studies, because here at Erie, we have a very strong genetic transformation lab. But through all of this history, whether it was in the field or in the lab, one of our main strengths is our people. And I'd like to take a moment to acknowledge the people that are involved in the root research that we're doing. So these are the people that are here as my live audience tonight for this lecture. And here's some photos of the work that they do routinely in the field. And I'm just so grateful to them for all their hard work. They never complain. They really like roots. And whether it's soil coring, washing roots, sectioning them, counting them, my staff is always very eager and dedicated to the work that they're doing. Another strength that we have here at Erie is a very extensive network of partners. So we are based in the Philippines, but we have country offices in many places in Asia and Africa. And we collaborate with different governmental research institutes, universities, other institutes. In fact, Erie doesn't release any rice varieties itself. We share the lines that are developed with partners, and they take it from there to go through the in-country screening and variety release process. However, in terms of root research, we have a less extensive history with partners. So here's some of the work that I was able to find a record of, some in Ubon in Northeast Thailand, some root studies in Bangladesh. And then on the right, there's some examples of work that I was involved with. We were characterizing traits related to the drought-tolerant variety Sapagidan. So this was at 2 different sites where we provided the soil core tubes and other equipment, and also root scanners, so that we could conduct some root studies at those sites. But over the years, that's one thing I had really hoped to do some more. And I'm happy to say that today I'll be able to present a little bit more work that we've been able to do with partners. Another interesting aspect of working at Erie as a root biologist is that roots are just fascinating. The rice roots, they're very distinct. So as you can see, they're always full of parenchyma, and that is, this root was sampled from an aerobic field. It was dry direct seeded. So you think of parenchyma as forming under flooded anaerobic conditions, but for some reason, rice roots tend to make a lot of parenchyma. We don't know why, but it's something very particular. And then the other thing is about the diameter. Rice roots tend to have very, very fine roots, and a lot of them. And because of that large proportion of fine roots, rice tends to make a very high root length density compared to other crops, especially in shallow depths of the soil. But so this has all made rice very interesting to work on. In addition, rice plants are adapted to such a huge range of conditions. So here you can see conditions ranging from dry and nutrient deficient in upland fields, moderately yield reduced and fluctuating soil moisture conditions for rain-fed lowland rice, and then water depths that are taller than we are for deep water rice. It's such a huge range of adaptation. And you can imagine the different types of soil that the rice plant experiences with these different kinds of environments. And this large range of adaptation means that rice really has a lot of genetic potential for adaptation to climate change. So one strategy for adapting to climate change that I'm going to talk about today is the management strategy of dry direct seeding. So many people are familiar with the typical rice paddy where the the field is flooded, the plants are transplanted into the muddy soil. But of course, that requires a lot of water. So one strategy to try to cut down on the amount of water that rice plants are using is to just sow the seeds directly into dry soil, just like a wheat crop or a maize crop. But because a lot of rice varieties are not adapted to these conditions, there can be a lot of challenges, and that's something we're working on. So two of the challenges that I want to focus on, that I have been focusing on with my research, and I will focus on during my talk today, are early stage drought, so seedling or early vegetative stage drought, and also micronutrient deficiencies. And actually this second one, I wasn't really thinking of. I was mostly just thinking about seedling stage drought. It was something I was very interested in. But over time, we realized after growing our rice in our experimental fields season after season under aerobic conditions and giving drought stress treatment season after season, our plants started looking worse and worse every season. And we realized that they were developing micronutrient deficiencies. But if we puddled the soil, brought it back to a muddy paddy field, then that problem went away and we could resume doing aerobic trials the next season. And it turns out that this is something that this micronutrient deficiency, this problem with aerobic soils, has been known for a long time. Going back probably 20 years at Erie, there was research on aerobic rice. And even then, they were observing something called soil sickness. So it's thought to be related to micronutrient deficiency. Maybe there's also some microbial aspect, but we have not characterized that yet. But not only do I see that in my experimental fields, but we're seeing this across some of our target regions, such as the Indo-Gangetic Plain. And one interesting thing that we have seen is that when we are growing the rice under aerobic conditions, the soil pH tends to go up and up and up, little by little over the season. So I think this may be one thing that is causing this problem or even exacerbating it. But not all rice fields are maintained under aerobic conditions for the entire season, so the pH can fluctuate just like soil moisture can fluctuate. But so anyways, I started out focusing on drought, but then it turned into also micronutrient deficiency because they're going hand in hand in these dry direct seeded trials that I'm running. So in order to help identify rice germplasm that could be useful for developing new varieties that are adapted to dry direct seeding, we have been screening a lot of diverse germplasm. And so, as I mentioned, one of the main resources that we have at Erie is the Gene Bank, which is hosting more than 130,000 rice accessions. And our group, over the years, we've screened at least a few thousand of these. And this is a list of some of the projects that we have done on screening gene bank material. Not all of them are necessarily related to dry direct seeding. Some of them were vegetative stage drought under rain-fed lowland conditions. Some of them were just root studies. But all of this screening of diversity panels helped us to identify some accessions that were promising, whether it was standing out for a certain root trait or for vegetative growth under dry direct seeding and drought. We have a lot of data, but at one point, we were just collecting more and more data. Meanwhile, my colleagues in the breeding department were setting up a new program on dry direct seeded rice breeding. So here are some of the features of their breeding program. They are basing their program on target market segments. So these are rice growing areas that have specific preferences, not just in terms of yield, but also grain quality, disease resistance, stress tolerance, many different traits. So based on all those preferences, the breeders have developed what they call product concepts. So these are long lists of all the different traits that they're trying to incorporate into the new rice varieties. As I mentioned, we have very strong partner networks. The breeders are sending out their lines across these networks for evaluation. They've also set up phenotyping hubs here in the Philippines, in Kenya, and in India, where they have more mechanization and more high throughput breeding strategies. They have a breeding scheme that is a very detailed strategy for how lines should be advanced, etc., and a plan for product release and scaling, which is basically identifying the best lines, getting varieties released, and then getting those varieties to the farmers who need them. But I'd like to take a minute to talk a little bit about this figure in the lower right. So I won't go through all these details, but this is the breeding strategy. And basically, what they're doing is elite by elite crossing, advancing the generations. Then they get to what's called stage 1. Those are the progeny of the crosses. Those get sent to multi-locations, and only the best lines from across locations at stage 1 are advanced to stage 2, which is also evaluated at multiple locations. And then that's where our partners take things forward by selecting what's performing best and most acceptable to rice consumers in their regions. But so the aspects of this strategy that I have participated in for my talk today include screening of gene bank material. So that lets us know about some promising lines, traditional varieties that could be used for what we call pre-breeding, because breeders in the elite by elite crossing are not crossing directly with land races. They're only using a closed pool of elite varieties. I was also participating in screening some of the lines that would potentially be used for parents. And then finally, I'll talk a little bit about my work on the stage 2 lines. But so as I was learning about this breeding strategy, I started thinking, as a physiologist, I've been screening a lot of diverse germplasm, but what's my strategy? As I mentioned, my group had screened many different sets, thousands of lines, but we were just doing more and more screening and seemed at one point not moving forward. So, oh, and before I move to that, a couple of other things about this strategy. One is that, as you can see, if you're going to work on characterizing a breeding pool, this is a moving target. The lines are changing every year. The breeders are moving fast. So that every time crosses are made, new sets of material, in principle better material, is coming through every single year. So because you're going to get a different set of material every single year, as for doing mechanistic studies, we need some kind of repetition. So because of that, there's a lot of importance on multi-location testing, not just in terms of having a range of environments that we're looking at things, but so that we can get some repetition of these lines, because next year it's going to be a set of completely different lines. So yeah, anyways, about the strategy, I wanted to have a strategy for my work too. And so mine is a lot simpler, but this is what I came up with. These were my preliminary stage gate system for contributing to breeding. So I started with having screened a lot of diversity panels. And from each panel, I selected lines that I thought were standing out for some trait that could potentially be useful for dry direct seeding. And then I put them together in one trial I called Best of the Best, where I took about 100 lines and looked at them all together in the same trial, which I had never done before. Now, in retrospect, I should have maybe spent a little bit more time on that, because I just put them all together in one season and it rained, so I didn't get very much drought. But I continued to screen a lot of the potential donors, these traditional varieties, over the following seasons. But then we took the best of the best lines and put them together with breeding lines. And so at that time, our breeding program hadn't set up yet their what they call core panel. They were still screening some of the most promising breeding material available at Erie, but that was what they gave me. And so, yeah, maybe that's one of the reasons I kind of skipped the best of the best part a little bit, or rushed it, was because I was so eager to finally start working on the breeding lines and putting them together directly with the land races in the same trial. And so for this one, what I called the gap analysis, we ran that experiment for 3 seasons. And it wasn't the same breeding lines that were, some of them were the same, but some of them were newly nominated by the breeders. But for all of those trials that we called the gap analysis trials, we did multiple different root measurements, including anatomy, root crown characterization, such as crown root number, mesocaudal length, lateral root density, and also we did scoring for root hairs. So I'd like to just say a little bit about how we got all this work done, especially for those in the audience that might be considering doing some high throughput root work in the field. I'll tell you about how we typically manage this. So as I mentioned, I have a big, amazing team. But you don't have to have such a highly skilled team like I do. I would suggest have a team at least to do sampling. It's nice that you can sample all the roots in a short amount of time so that you can be assured that there's not a lot of variation among your samples due to the time that it took to harvest. But even with a big team, we do a lot of preparation. We never do any labeling in the field. We prepare all of our sampling containers, all of the labels. We don't write anything. We print it all out. Everything is prepared ahead of time and organized very well to save time so that we can sample very quickly. And then for today's talk, I'm going to kind of focus on root hairs because that was one of the traits I was really interested in and saw some interesting results. And those of you who have worked on root hairs know that it can really be a headache to try to quantify the length and density. Usually we take an image, look at it under the microscope, count the root hairs within a given area. But it can sometimes be very difficult to see the root hairs. But so the approach that we took was to use a root hair score. And that was something that I had picked up from one of my former lab mates from the Lynch lab. And the scoring system I used was very simple, 0 to 4. So yeah, 0 being no root hairs, 4 being long and dense. So all I had to do was look at the root and give it a score. And for this scoring, another important strategy was to use digital data collection. So here's a screenshot from my Fieldbook app, and I love this app. It saves so much time, and I highly recommend it to anyone, especially students who may be in the audience. You shouldn't be writing with pencil and paper. Trust me, you can just, all you have to do is prepare. You put your experiment layout into the app, and then you can go through the samples very quickly. And so the way that we did this is one of my staff members. She had prepared the roots. She dipped them in the stain and put them in a petri dish, and then I put them under the microscope. And with that strategy and using the Fieldbook app, every Wednesday from 10 to 12, we met up in the microscope room. And over the past few years, I was able to score more than 10,000 roots. So just by having these kind of high throughput things like a scoring system and using digital data collection. So I just wanted to mention that in case it could help encourage others to move towards more high throughput root phenotyping in the field. So as we went through this root phenotyping, there were some interesting observations. First of all was the huge variation in nodal root diameter. And definitely I expected that from the gene bank accessions, but I didn't realize how the breeding lines would be so variable. So here's just 2 extremes, one very small diameter, one large diameter, but yeah, these are breeding lines. They're not diverse material. And when you look at them above ground, they don't look all that different. Another surprising thing was the root hairs were not straight. I think a lot of us are used to root hairs maybe coming from paper roll-ups or from agar plates where they grow very straight from the root. And we did see some of those sometimes, but in general, they were very wavy, which also just reinforced, I was glad I decided to score them rather than trying to measure them. Another interesting thing was that in some trials, there was a huge difference between the root hairs on the lateral roots and the root hairs on the nodal roots. So you can see these two images. The one on the left, I kind of tried to focus more on the lateral root, it has a lot of root hairs. Whereas on the right, I tried to focus more on the nodal root, the larger diameter root. It doesn't have many root hairs at all. And I think that tended to be from the trials where the stress was more severe. So perhaps the nodal root grew first when the stress wasn't that severe, but when the stress came very quickly, then the lateral roots grew and had a lot more root hairs in response to the stress. At least that's my guess. And then finally, and this is something I think is really important, root hairs often proliferate when there's a problem. So this was something we observed long back from one of my students working on dry direct seeded rice. She saw that every time there was some bump, like a gall forming on the root, it was really, really had a lot of long root hairs. And so she went on to do a lot more characterization on root knot nematodes, which is a common problem under dry direct seeding in rice. So yeah, those are some of the interesting things to start off with. And as I mentioned, we're focusing on drought and micronutrient deficiencies, but our performance indicators for these traits were, first of all, I used relative growth rate for drought. And the samples that we took for relative growth rate were at the end of the seedling stage drought and then 1 week after rewatering. So this is actually more of a drought recovery measurement than a drought tolerance measurement. And the reason I really wanted to look at relative growth rate was because if we just looked at, for example, biomass, it might not represent drought stress tolerance very well because in diversity panels, there's such huge differences in biomass. So that's why I preferred relative growth rate. And also because I think these fluctuating soil moisture conditions are really much more representative of the type of drought that's occurring in farmers' fields than just a progressive dry down. And for the micronutrient deficiencies, we just took chlorophyll concentration index. So we use this instrument that logs the data. And I'd also like to say that across our trials, so each of these trials had between 100 and 200 genotypes. We grew them under 3 reps. But the stress severity was variable. In some of the trials, it rained and they didn't get very drought stress. Our second trial, you can see here, also had really severe, what we could call soil sickness. That was in a dry season. The next trial was in the rainout shelter, so the drought stress was also pretty severe. And then finally, we had another trial in the open field that also received some rain. And so with this range of different levels of severity, both in terms of water and micronutrients, you can imagine it might be difficult to find traits that are consistently correlated with what we considered good performance under dry direct seeding. So I looked at different correlations of the different root traits we had measured, and as you would expect, very few of them were consistent over multiple trials, or if they were in opposite directions. The only thing that we observed to be correlated with relative growth rate over multiple seasons was crown root number, which was negatively correlated. And so because of this, I wanted to find another approach that could kind of encompass how all the different traits are interacting with each other, and also with the type of environment that they were exposed to. So that's why I got really interested when I learned about this concept of integrated root phenotypes. So what are integrated root phenotypes? These are combinations of root traits that explain crop productivity as identified by multivariate clustering. And individual root traits have different effects on plant growth depending on the expression of other root traits. So that's what this idea of integrated root phenotypes takes into account. And to my knowledge, this idea came from the Lynch lab. So here's a few papers, all from the Lynch and Brown labs, looking at different integrated root phenotypes in response to stress. So that's what I wanted to apply to all the root data that we had here for dry direct seeding and rice. So what I did was I ran the script from the Klein et al. Paper, and I looked at first relative growth rate. I chose what were the clusters that were performing best for relative growth rate, and then I looked at all the different root traits that also stood out for that favorable cluster. And so here's the list of different trends across the favorable clusters for relative growth rate. But again, I wasn't seeing much that was very consistent, but that's okay, because these are, as I mentioned, these are different environments. Although if I looked at just the most severe trials that had the most severe drought stress, those had consistently, the favorable clusters had high crown root number and a high L-type lateral root density. So then I also did the same for chlorophyll concentration index. And in some cases, like this example, you can see there were actually two clusters that were both performing well for chlorophyll concentration index, but they had completely different root traits that were related to them. So then, oh, thank you so much. When I looked at the traits related to those favorable clusters, in this case, we did see some consistent traits. So for chlorophyll concentration index, both root diameter and steel diameter, whenever they stood out, they were higher than in the other clusters. And then for two of the traits, metaxylem diameter and nodal root hair density, in most of the favorable clusters, they were high, and there was just one exception of a cluster where they were actually lower than the other clusters. So another observation that was interesting is that when we looked at what these favorable clusters were comprised of, they were actually a mix of both breeding lines and the potential donors or the gene bank accessions. But the lines that were identified in multiple favorable clusters over multiple seasons were the land races from the gene bank. So actually, I've been able to recommend those to feed into the breeding pipeline. And that's using what we call the trait development pipeline. So my colleagues are now making crosses with those gene bank accessions in order to develop populations, do some genetic mapping, and hopefully identify some genetic regions that are related to either chlorophyll concentration index or relative growth rate under drought. And then those genomic loci will be introgressed into elite backgrounds. And those, what we call elite donor lines, can then be used in the elite by elite crossing and phenotyping. And then by that time, when we had gone through what we called the DSR gap studies, by then the breeding group had set up what they called the DSR core panel. So this is what they plan to use as their closed pool that they would be doing elite by elite crossing with. And so because that set was sequenced, we also did a number of measurements there. So we, across those, all of those entries, plus some of our potential donors, we measured again relative growth rate, chlorophyll concentration index, crown root number, and also nodal root hair density and length scores. So at first, my idea was maybe those traits would be always present in favorable clusters, and then we could find a genomic region for that, but it didn't turn out that way. but it will still be helpful to be looking at these traits and the genomic regions that are related to them, especially once we know what can be contributed from the potential donors that is not already present in the core panel. One comment I would like to point out is if you look at these distributions, the breeding lines are good. We don't really have much from the potential donors that are outperforming the breeding lines for any of these traits, with the exception maybe chlorophyll concentration index. And the reason why I think we have some lines that are performing better than breeding lines is because in many of our trials, like I said, we give this repeated aerobic conditions, we're getting micronutrient deficiencies, and that's not something the breeders had exposed those lines to before. So it really pointed out to me that, you know, as physiologists, as root biologists, if we want to contribute to breeding, we need to know what it is we're trying to contribute to. We need to know our target. And then it's better if we can work in a way that's complementary with the breeders. So in this case, I was focused on seedling stage drought, and it wasn't overlapping with the breeding group because they knew that's what we were interested in doing. And so we just shared information. So from this screening, maybe it will present some interesting root traits that will potentially be targets for selection. But for sure, we've generated plenty of data, not just on these seedling stage traits, but also agronomic traits coming out of these trials: height, time to flowering, grain yield, biomass. And that has been used by the breeders to help them make their decisions in what to cross. And again, this is their strategy. So the lines here, this core panel, that's here at the top where they're doing this parental selections and elite by elite crossing. So that was the work we had done as the breeding program was getting set up. So, and as I mentioned, after the crosses, then the lines move to stage 1, get selected, and then stage 2. And this year was the first of the products coming out of the elite by elite crosses that were set up specifically for this direct seeded rice breeding program. So I was really excited to start looking at those stage 2 lines. And finally, I had the opportunity to do a lot more extensive root work at our partner sites. So these are the partner sites where we've been working. We have one project funded by the Gates Foundation on developing new varieties adapted to dry direct seeded conditions. So these are the sites where the lines were growing. All of this was planned by the breeders and the partners. My group didn't have to do any planning or implementation of this. But the advantage is that many of the same lines were growing in all these different places. So there's my repetition. So next year, there will be a totally different set of lines. But this year, we were able to go to here. This is 6 places. We had an extra trial in Hyderabad, and then I had an 8th trial here at headquarters in the Philippines. So yeah, as you can imagine, I was really excited about this because after all the work here at headquarters, you really want to see what are the plants doing in the target environment. And it's just so great to meet with these partners. I have a lot of respect for them. They are true experts on what are the needs of their regions, how the rice plants adapt to their regions, what are the conditions and the reasons where they work. And they also, I learn a lot from them. Every time I visit, I take pages and pages of notes. So as you can imagine, it's really a gratifying experience to visit these places and learn from them. But I have to say, I was just excited to collect all the roots and then finally get to look at them under the microscope because I was so curious about what is happening underground. So what I did was I just visited all of the places, and then the last place I visited was one of the Erie hubs in Varanasi where they have a nice microscope set up. So the last day of my trip, I was able to very quickly score these several hundred roots that we had collected under the microscope. So what did we see? Basically, one of the things that really stood out was just huge variation among the different sites. And ranging from Varanasi, which for some reason had hardly any root hairs, up to IRI in New Delhi, and also here at IRRI headquarters. For some reason, that season, our plants had really a lot of root hairs. They were long and very dense. And again, this is the same material across all these different sites. The environmental effect was huge. And interestingly, I also collected chlorophyll concentration index at the same time as we collected the root samples. There was a negative correlation between the chlorophyll concentration index and the root hair density. Also a negative correlation with root hair length score. I was a little bit surprised about this, but to me, I think this represents how in locations that are stressed, root hair formation is induced. So in these places where the chlorophyll concentration index was lower because of soil-related stresses that we didn't quantify, well, we have a little bit. We've measured soil pH and different nutrient levels. But because of those stresses, the plants weren't able to, that's reflected in a lower chlorophyll concentration index. And those were the places where the roots were making more root hairs. But then in terms of genetic differences, there was really a big range. So we had the line I have here in green, this is Sabhagidan, this was our Czech. This is one of IRRI's first developed drought-tolerant varieties. It's still very popular in the Indo-Gangetic Plain. But you can see these stage 2 breeding lines, some of them had higher root hair density than our Czech, some of them had lower root hair density. But this Czech itself changed a lot from Varanasi up to some of the other sites. So I thought that was interesting. And to me, again, I feel like this is the root hairs being expressed when the environment is more stressful. In contrast, we had another line, also considered as a Czech, ISRR-176, and it was a lot more stable. ISRR-176 is also known as Katihan 2. It was released in the Philippines as a variety specifically for direct seeding about 10 years ago. So you can see this one is a lot more stable across the different environments. And in fact, it maintained a higher chlorophyll concentration index. So one thing I'm thinking is, in some of these traits that are induced by stress, yes, the expression of that trait is a strategy for the plant to adapt to that stress, but it could also reflect that there's some problem. So maybe it's a good idea, if we see that happening, to look for more stability. So if it's one thing if the plant is making a lot of root hairs when it gets stressed, but if it also makes a lot of root hairs when it's not stressed, then maybe that means that plant is better adapted to different conditions, and it's not just making root hairs because it has some problem. Okay, so in conclusion, yeah, I've really learned a lot from this experience. Seeing what the breeding lines are like, being able to work with partners, seeing how the plants, the same genotypes, responding so differently across different experiments at different sites. So some of the things I've realized is that, first of all, it's important to know your target. So in my case, my target is broad. There's so many breeding lines, and it's a moving target. Like I said, every year things change. So we have to try to keep up with that. Another important point is that breeding lines that are selected for a given environment may be hard to beat. So even though breeders are not selecting for root traits, at least in this case, they're still selecting, and they may be indirectly selecting for root traits. It's likely that they are. So we have to keep that in mind, that we can't just walk in and say, oh, I have this great line or this great root trait. You have to know, what is it that you're trying to improve? Maybe the breeding lines already have that trait, maybe through the breeding that was done historically. But now if you're in a different situation, maybe you're a regional breeder or working with a regional breeder who has one specific variety that they want to improve. Say they have a variety that's super popular and they just want to make it more drought tolerant, or they just want to make it better adapted to dry direct seeding, that's a lot more defined target. So it's not changing over time, and maybe you can more simply introgress something that would benefit that variety. In terms of integrated root phenotypes, I've learned that a given trait has different effects depending on the other traits expressed. And that the expression of some traits, for example, root hairs, may indicate stress susceptibility rather than an adaptation of a given genotype. So for those reasons, I feel very hesitant to recommend just a single trait for anyone to select for, especially for the breeding program here, seeing how much huge variation there is among root traits among the breeding lines. And finally, I hope that I've been able to illustrate how trends across experiments with a range of stress levels may help indicate the level of adaptation of a given pool of genotypes. So like I was saying, especially in the case of root hairs, some traits may be just reflecting a degree of stress. But if the trait is more stable across different environments, maybe it is conferring some advantage in terms of adaptation. So that's everything I wanted to share with you today. We haven't published any of these results yet. We're still working on summarizing the data. We hope to do some genetic analysis. So any suggestions would be really welcome and also appreciated. And finally, I'd like to share some acknowledgements. Of course, as I mentioned, I thank my staff for all their hard work and dedication to roots. I'm very thankful to the partners that we work with. They're really representing the farmers, and that's our ultimate target. And I'm especially grateful to the breeders who have helped me so much over the years and have been really great to collaborate with. Thank you very much.
Thanks, Amina. That was really great. A really great example of how difficult it can be to screen roots in the field and in large numbers like you're doing. So thanks for that inspiration, I guess. So we have some questions in the chat, and there's obviously lots of applause. So let me just see what we have in the chat just now. So I mean, maybe the best way is for the people just to unmute and ask their questions. So Glyn was first. I don't know, Glyn, are you happy just to ask your question to Amelia?
Yep. Hello. Hi. I'll uncloak.
There we go.
That's a great talk. Thanks. I was wondering how you characterize the soil physical environment. I know you're doing a huge experiments, but how do you measure how dry the soil is or how hard the soil is, as obviously that can have implications for roots?
Thank you. Hardness, I haven't worked on hardness so much, although we took bulk density samples at each of the sites where we worked in India and in all of our fields here at Erie. So that's, I think, basically the only kind of hardness physical measurement that we have. But we've used tensiometers in all of our trials, and actually that's something I've put a lot of effort into, providing tensiometers for all of our partners, both in South Asia as well as East Africa. And for me, that's probably my preferred measurement. Before, when we were working in lowland rice, people hated the tensiometers. They didn't like it because in lowland rice, the soil cracks and then the tensiometer stops working and people give up. But for dry direct seeded rice, the soil doesn't crack so much. And so I hope that people will be more encouraged to use it.
Yep. So tensiometers seem like a really useful measurement of soil suction. So, and I wonder if it may be possible, and Adam might have ideas, for instance, of relating that to the soil strength characteristics, so you might get both from, I don't know. Sorry, thank you.
Okay, thank you for your suggestion.
Thanks, Glyn. Leah, are you happy to open your mic and ask your question?
Yeah, hello. Thank you for a great lecture. I was just wondering, you had mentioned, I believe, if I got the number correct, you had upwards of 130,000 accessions to choose from in your gene bank to screen for sort of genetic root diversity. And that's quite a difficult task, because how do you capture a wide range of the genetic diversity? But obviously that's a somewhat impossible task with high throughput root phenotyping. So how did you select those accessions that you did end up screening?
Actually, most of the selections were selected by my colleague, Ken McNally. So in some cases, we wanted to focus on what's called the Aush subgroup. So not indica, not japonica, but Aush, because those are lines that have evolved in Eastern India and Bangladesh and very stress-prone regions. So that was one reason. Another reason is there's a set of 3,000 rice varieties that have been sequenced. So that was another set. How those exactly were selected, I don't know. And then another, we have a collaboration with New York University with Michael Perugunam's group. And so he was the one that selected another set. I wasn't the one that selected them. And that's another good point, is that all this screening, it was done for so many different reasons. Some of the projects were, trying to just do screening for identifying lines, but others were completely kind of different goals. Like NYU, they were doing RNA-seq and looking at evolutionary questions, but it was still a great way for us to get some screening done in the field. So that's why in the end, we were able to put all those things together.
Thank you. I really appreciate that. It helps me a lot. Thank you.
Thanks, Leah. Davide Bulgarelli, would you like to ask your question?
So I would like to join my colleagues. So thank you very much for your talk. So my question is, I wonder whether you have a look at the level of colonization from mycorrhiza on your lines, and whether eventually there is a trade-off between root hair formation and mycorrhization, in the sense that if the conditions are conducive for the establishment of this interaction, maybe plants are not developing root hairs, they go for mycorrhiza, and probably this is more efficient in scavenging micronutrients or resisting to drought. I wonder whether you have looked at or you are planning to look into this direction.
Thank you. Yes, that's a great question, because our next speaker will be Emily Cervante, who is here visiting Erie, and she is characterizing the mycorrhizae in some of these same trials where we did the root hair measurements. So I can't answer your question if there's a trade-off, but I hope soon we will be able to answer that question.
Thank you.
Thanks, Davide. I have a follow-on question to that in a sense as well. The root hair length of rice is quite short in comparison to other species. So we work on barley, as you probably know, and the root hair length in barley is much greater than in rice. Do you think what's the capacity to breed longer root hairs into rice, and do you think you can achieve root hairs equivalent to those you get in wheat and barley, for example, which may be a millimeter or a millimeter and a half even?
Yeah, that's a good question. I mean, if we're not seeing the genetic variation anywhere for longer root hairs, it would be difficult to introgress it, but maybe through some transgenic or genome editing approach, we might be able to overexpress something that could elongate the root hairs. I don't know. But again, I think we should take this question seriously about trade-offs. And could there be a trade-off of having really long root hairs in rice? Because why doesn't rice have long root hairs already?
Mm-hmm. Great. Hari Gopala Krishna, would you like to ask your question? If not, then I can relay it, which is they wondered how the soil pH changes during the different stages of the crop. Is there a large variation in the developmental stage in soil pH, for example?
Well, from what we were seeing, it was not so much stage specific, but more specific to the soil moisture at the time when we were taking the measurement. It seemed like the longer we were having the soil aerobic, whether it was within that season or across successive seasons where we were growing same trials, the soil pH tended to go up. But yeah, I can't say that I have monitored, we haven't monitored the soil pH that intensively. We just started doing this when we realized about this micronutrient issue and wanted to try to figure it out. When when we were taking the measurements at partner sites, we also measured the soil pH there, but that was just at one time point in the study.
Great. We've got time for more questions, so we'll continue if you don't mind. Bojana, would you like to ask your question?
Sure. Can you hear me?
Yep, perfect.
Okay. I don't know what's happening with my camera, but for the talk, I'm really with it. My question actually is in the same line as Giambo's, coming one further down. Nutrient availability and water, they sometimes go together, right? And I was wondering which specific micronutrients cause the problems in your soil, and do you see repeatability between the various trials that you performed, like in the example of the Indian multi-trial example that you showed?
Well, typically, it's typically thought that these are both iron and zinc deficiency problems. And I can't say which is more. We haven't done that much quantification of the leaf nutrients. We did here at headquarters some, and we saw a correlation between our chlorophyll concentration index measurements and the leaf zinc content. So those are the main micronutrients that we're focusing on.
All right, thank you.
Just to add to that, the work we do on barley again is looks at manganese deficiency, and that's a big problem for, well, it has a massive effect on the chlorophyll function because it binds the photosystem 2 proteins. So manganese is very strongly related to zinc as well. So there'll be correlations there, I'm sure.
Okay.
Cool. Adam, would you like to ask your question?
Hi, Amelia. Nice to hear you again and see you again. The last time I was in Erie, can't remember how long ago that was, you were doing a lot of work with a pressure bomb. And I think you were looking at hydraulic conductance. So I wondered whether or not what your thoughts on that are as a trait, because we have definitely got some interesting results on hydraulic conductance for drought in rice.
Yes, thanks. Yeah, we did that. And actually one of our speakers this session, Alex Grondin, he also really focused on that a lot during his postdoc here at Erie. And we saw a lot of genetic variation. But actually, well, we worked with the pressure bomb, and then we did really a lot with this sap bleeding rate, where you cut off the shoot and then collect the sap that comes out of the root zone. And I would say that trait probably, so we were asked to consider stage gates, like thinking about our work and what can move forward. And because bleeding rate, in some cases, the drought tolerant lines had a higher bleeding rate. In some cases, they had a lower bleeding rate. So I was feeling like it was kind of variable. I know that sounds kind of funny as a root biologist because everything is variable. But because I wasn't sure if high or low was good, and I'm still not completely sure, maybe it depends on the growth stage. I didn't really continue that through this work on the dry direct seeding.
Thanks, Adam. Jambo, Bojana asked a part of your question, but I mean, you have a second part to your question. Would you like to ask that, Jambo?
Yeah, yes, thank you. So my question, it's very difficult to separate the drought with phosphorus deficiency. Normally under the drought, the water shortage is a big problem, and have a big effect on the diffusion of phosphorus and also zinc. So that means when we talk about the drought and how to separate the effect of the water deficiency and also zinc or phosphorus deficiency. So any comments for this? Okay,
yes, thank you. We haven't been focused so much on phosphorus deficiency. In the soils where we're working, especially in India, fertilizer is subsidized, and the soils are actually quite fertile in terms of phosphorus. I'm not working in what we consider phosphorus deficient soils. And the other interesting thing is these micronutrient deficiencies that we're seeing, the iron and zinc, it shows up in any trial, even ones that become flooded or have standing water. And I think it's maybe more complex than just water availability. I think, as I was mentioning, a history of repeatedly growing the crop under aerobic conditions, rice is very sensitive to this. And I don't understand everything that is happening in the soil to cause this problem, but it's widespread. And it's not completely related to what we would call drought. I think it's more related to long-term aerobic conditions of the field. But I think we have a lot more to learn.
Okay, thanks.
Thanks, Jambo. Ken, would you like to ask your question?
Yeah, thanks, Amelia. Interesting talk. I was just wondering about Erenkeimer formation and whether this differs when, or what the response is under direct drill conditions, because obviously, yeah, Erenkeimer can be formed by a natural development response or in response to environment. I was just wondering whether you did any quantification, for example, of Erenkeimer and how this linked to CCI, and whether there's a beneficial trait for Erenkeimer formation under direct drill. I think it goes a little bit because there could be potential links between Erenkeimer and root mechanical traits, which may allow it to overcome harder soils and drought conditions, as a hypothesis.
Okay, yeah, thank you for that comment. I mean, it's really surprising how much Erenkeimer we see, even in trials that are never anaerobic. The rice roots are just constitutively forming a large amount of Erenkeimer. So in the past, we have quantified that. We're also quantifying it a lot now for other projects. For example, we have one project related to greenhouse gas emissions where we're doing a lot of quantification of Erenkeimer. But honestly, I can't really visually look at a root that came from a dry direct seeded field and a root that came from a flooded paddy and tell you which one is which, because it's that much Erenkeimer coming out of the dry direct seeded field. One interesting observation we've seen, though, is that many times at a location of the root cross section where a lateral root is emerging, there is no Erenkeimer. So you can imagine that could have some implications for at least gas flow through those Erenkeimer. But maybe it means that we could kind of estimate porosity just based on lateral root density. I don't know. But it's just, for me, it's just really surprising how much the Erenkeimer there is all the time, regardless of the soil moisture.
Yeah, establishment. Yeah, interesting comment about the lateral roots and no Erenkeimer around it. It's quite interesting though. I did some laser ablation stuff as well, and it's quite interesting that I wonder whether it's because of a strengthening mechanism in the cortical tissue from the lateral root passing through the cortex from the steel. So I wonder whether that actually strengthens it, and therefore you don't get Erenkeimer formation near lateral root development. Just an idea, but yeah, an interesting observation as well. Thank you.
Thank you.
Thanks, Ken. One last question from the chat. So Dennis Florentin, would you like to ask your question? If not, then basically Dennis was curious about whether the planting design had an impact on micronutrient acquisition. So he says, we know from previous research that plant roots might change the rhizosphere more easily at high densities. So whether the planting design had an impact.
Yeah, I think that's a good question. And density is really a hot topic in rice. We're trying to decrease the seeding density for many reasons. The cost to the farmers. In some places, the seed source is contaminated with weeds. So we're trying to, our agronomists here are trying to really reduce the seed density. I just remember a study done by Matthias Wisua long back, where it was a zinc deficient field and the plants were really hating it. They looked so bad. But in rice, a lot of times after we transplant, we leave a little bundle of the extra seedlings right at the front of the plot in case you have to fill some gaps later, in case some snails eat the plants or whatever. That little bundle looked so good in his trial. I saw some photos. So all I could conclude was there was some kind of rhizosphere, maybe nutrient solubilization or something happening there. So yeah, I do think that how densely things are planted really could make an effect. But yeah, I mean, from us, from the big picture, we're trying to reduce the density.
Yeah, it's complicated, right? So if you could just indulge me with one final question then. If you had one wish and that wish was to develop a particular phenotyping tool, what would that phenotyping tool be as far as roots are concerned?
Well, the first thing that comes to my mind is that we still spend a lot of time on the anatomy. I know that there's the laser ablation that helps a lot with the cross sections, but we haven't been able to get any of the automated analysis software to work very well on our rice roots. So we're just still having our interns manually analyze hundreds and hundreds of images. So yeah, that would be my dream, if someone could develop an application that works very well on the anatomy.
If that happens, let us have it as well, please. That'd be great. Thank you. Okay, so Amelia, thanks ever so much for that. It was obviously a very stimulating talk. We got lots of interest and lots of questions. So now we come to the part of the day where we hand over the award. So I'll hand over to the people in the room of the Philippines, where we have Dr. Sankalp Bosale, who's the Interim Director of the Rice Breeding Innovations Department, IRRI, is on hand to present Amelia with the medal. So over to Dr. Bosale, please, for the award.
On behalf of IRRI, I would like to again say our big congratulations to Amelia and the entire team that has helped her, for years now to decipher the complications that you have all heard about. Also praiseworthy is the team's engagement and effort on bringing the upstream research to fruition through effectively finding pathways to integrate that in a product development pipeline. So I mean, as upstream scientists, we always are challenged to find the larger motive to integrate our work and see the light of the day. I think Amelia and the team at IRRI, supported by IRRI's strategy, has really just made that. So I hope today's talk was inspirational for all of us online and the colleagues who've been really asking very valid questions. I also think institutions such as CGI or IRRI particularly, through its partner networks, offers a unique opportunity for all of us, including those who are online, to collaborate to achieving our basic overarching goals of achieving food, nutrition, and income security for the Global South. So I welcome you all to continue to explore the collaborations with Amelia, the team, and the broader group of colleagues we engage with through our national networks, and would like to really thank you for the recognition of the efforts of IRRI through Amelia and the team by receiving such a prestigious award. I think, again, my congratulations and to you, Amelia, and thanks to everyone who has been involved in making this possible. Thank you. Really appreciate having this opportunity to present this award on behalf of IRRI's Rice Breeding Innovations team. Yeah, thank you again. Appreciate it.
Thank you, Dr. Bosale. Amelia, I guess you're probably overwhelmed by the medal just at the moment because it is very special. Is there anything you'd like to say?
Well, again, to the Dundee Root Medal Committee and the International Society of Root Research, I really appreciate the recognition and this chance to acknowledge my staff. And I hope that the work we presented could somehow be helpful to other root scientists that are interested in doing field work as well.
Absolutely.
Thanks, Tim. And welcome back, everyone, after the little break for workshop session number 1. And we're staying in the Philippines, and we're going to start with Emily before we move over to Helena, and then end with Alexandra in Senegal. So Emily works at the University of Cambridge and currently visiting IRRI and working with Amelia. So over to you, Emily. And just to give you a warning, I will try and keep you to time. So I'll turn my camera off, and then hopefully someone in the audience, because you might not be able to see me, will notify you when I put it back on. And I'll do that with 2 minutes to go to give you a little bit more advance notice. So over to you. So thank you very much, Emily.
Hi, everyone. I'm Emily Savanti. I'm a postdoc in the Crop Science Centre at the University of Cambridge, working with Uta Puszkowski, but I'm here in the Philippines as part of our collaboration with Amelia's group. And I'm going to talk today about our work in assessing the potential of arbusca mycorrhizal symbiosis in aerobic rice. Yeah, so I'm at the Crop Science Centre, which is a building that's part of the University of Cambridge. And one of the main goals of the centre, we have multiple group leaders working on sustainable nature-based solutions to crop nutrition. So fertilizer use and production is a massive contributor to global greenhouse gas emissions, and we're hoping to use more sustainable already in the soil solutions to reduce this. And as many of you know, in natural ecosystems, plants have they engage with beneficial fungi and bacteria in order to gain the nutrients from the soil that they need for growth. And in the breeding and in modern agriculture, we haven't really been selecting for these kind of symbioses because we add high amounts of fertilizer, we till, which breaks a lot of the networks of these microbes, and we add fungicide, which in a lot of cases will actually remove them from the soil. So we aim to look at how we can bring their use back in agriculture to try and use them to reduce application of fertilizer. So I think a few people in the audience will already know about arbusca mycorrhizal fungi, but as an introduction to anyone who doesn't, they're a form of mycorrhizal fungi that live within the soil of plant roots, and they're characterized by forming these, what I think, really beautiful arbuscles that look like trees within the cells of the plant root. And here they're stained blue. That's how we do a lot of the quantification, so they're not actually blue, but we can see them within the plant, and that's where they really complete their life cycle in this mutually beneficial symbiosis. So the plant will give away carbon to the fungus to help it grow, and in return the fungus will give more nutrients to the plant, like phosphorus. So they're more and more considered as natural biofertilizers that have the potential to be brought back into more sustainable agriculture. So as I said, we work on mycorrhiza, and as do a lot of people, and decades of research really has gone into the genetic mechanism, how the plant and the fungus are interacting. And we know that there's this potential that the fungi can help the plant increase performance, especially under low nutrient, also a lot of abiotic and biotic stress tolerance, helping carbon sequestration by forming these massive networks in the soil, and also helping soil aggregation and improving soil health. So they have the potential for massive benefit. But and we saw this quite recently in work from Brewery Sawyers at Penn State, where he showed maize plants in the field in a median input system, and he used genetically restricted this AMFR on the right plants and compared to non-restricted plants that could engage with the fungus, and we really see this difference and the potential that the fungus has in contributing to really healthy plants in the field. So what we also know is that this relationship is genetically driven, and the plant genetic background actually has a big effect on how colonized the plants are and how much they respond in terms of performance to them. And there's already been work done in rice on the rice diversity panel, and also in maize and in many different species, showing this genetic variation. And we've also more recently seen variation even within genes that we know are related to the symbiosis, and that itself causes changes in colonization, and it also can contribute to that difference in how the plant is responding. So we now come in with the big question of how can we know that the fungus can do what it does naturally for a lot of plants in nature and help contribute to fertilizing plants, and actually therefore boost performance, and which cultivars do we need to use for that to work. So this is perfect timing, as Amelia said, and that's why we're working here with IRRI. There's a big push towards direct seeded rice, and especially dry direct seeded rice gives the opportunity for aerobically grown rice, which is perfect for us because the fungus doesn't like water. It will not grow or persist very well in anoxic conditions such as a waterlogged paddy. So we're perfectly at the right time to come in indirect seeded or any kind of water saving rice cultivation and look at the potential for this fungus in those systems. So I started with my PhD project a few years ago, and we collaborated with IRI to use the 3000 Rice Genomes project, which hadn't yet been looked at for AM traits. As I said, there's already other work in different diversity panels, but this one had not been investigated. So we first started by making a sub-panel purely of indica to make it easier in terms of how many we can look at, and also power of the GWAS. And we created this panel of 200 cultivars, still maintaining the global representation that is within that whole indica panel in the whole project, and all the subpopulations within indica. And then we interrogated this in a glasshouse investigation in Cambridge, where we had plants that were non-inoculated and plants where we inoculated with the fungus. And this is shoot biomass response to the inoculation. And you can see we again have this huge variation in how the plant responds, anything from actually a negative effect all the way to a positive, at least for biomass and growth. So again, we're seeing that genetic variation coming back. And we also looked at the amount of colonization in the roots. And to do this, we used a foliar marker developed by Ian Baldwin's lab in Jena in Germany. And this is a metabolite marker that is only produced when the fungus is within the roots, at least in rice. So we used this, we measured the amount of that in the shoot, which meant it was a lot more high throughput than the usual root phenotyping we would do. And again, you can see that we saw this massive variation in the amount of colonization in the roots between those 200 different lines. And we have performed GWAS on a lot of the traits that we measured, and have now identified quantitative trait loci associated to how much fungus is in the root, and also this kind of response measure of how the plant is reacting to that. And we're now in my postdoc looking at candidate genes, and we found some transcription factors that we're really interested in because they're associated to nutrient response, and some cytochrome P450s just as a teaser, but we're continuing to look at these kind of genes. But we're excited that we have these loci that are really contributing to understanding where that variation is coming from within rice. But we also want to ask, okay, if a plant or a rice plant is responding well to the fungus, how consistently does it do that in different locations in the glasshouse over multiple seasons in the field? And so I have, from the 200 indicas, created a smaller panel that we then look at in multiple locations and in the glasshouse. So we have collaborators, and we've been working with Amelia and here with the team at IRI in the Philippines. We also work with Dr. Mavis Osai Owasu in University of Ghana at WACI, and we're exploring new collaborators in South America as well to really put the same lines in the field with inoculation with the fungus and see if we can have these top benefiters that really love the fungus, or is it going to be more dependent on the environment, which we often see in the field. So as an example, this was a glasshouse experiment that I did in Cambridge in the glasshouse, and I inoculated with our model Rhizophagus fungus, so a single species. And this is the sub panel of indica that we chose. And as you can see, we have some plants like Bongzen that really love the fungus. So you can see the difference between the non and the inoculated plant, that the inoculated plants are much bigger, and this is all measured by biomass at this stage. And then we have plants like Chama, which really don't care whether the fungus was there or not. The biomass is the same between the inoculated or non-inoculated plants. And so we picked these out as lines that we're interested in. And then what is also good to see is that that variation that I saw in that initial screen of 200 lines is coming back again in another glasshouse experiment, this time actually at a later stage in development. And then I performed another, well, my master's student actually, Celia Watling, contributed a lot to this. She did another screen, this time with different species of Arbusca mycorrhizal fungi and that's what you see in different colors compared to the red, which is the non-inoculated. And if we pick out those same lines, again, CHAMA doesn't really seem to care whether the fungus is there or not, regardless of the species. And then those favorite benefits from before, in some cases, they are showing this increase in biomass in inoculated with different species, and in some cases they're not. So we can extend that further, that this genetic variation is dependent on the plant background, also the AMF species, and actually probably the inoculum source, because you might have noticed that some of the rhizophagus plants are not coming back with what we saw in that first experiment, and this was actually a crude inoculum compared to a clean. So I think that is also affecting this. So again, we have that question of consistency, and are some of these ones that seem to be coming back consistent also in the field? So we moved to IRRI, and thanks to the whole team in this room who have helped with these experiments. So we perform inoculation experiments in the field in the Philippines, where we have plots where we can't get rid of the fungus. We don't want to sterilize the soil, so we have just plots with native, probably fungi, and then plots where we add inoculum as well and compare the biomass. And what is quite striking is that when we looked at the early vegetative stage, actually the inoculum had a negative effect on everything, which I then looked in the literature, and this has been seen before. And maybe it's to do with too high amounts of inoculum or too soon, or the fungus is kind of coming in at the wrong time. And then at harvest stage, we again see these lines coming back where then sometimes they don't mind if the fungus is there or not, and sometimes they're really boosted by it. So again, we see genetic variation, and that's coming back in the field as well as in the glasshouse. So we continue now to try and test these cultivars over multiple seasons and in different locations, and see if some of these favorite lines are actually consistent, and if we can tease out the reasons why we see that different response. We can also do that genetically because we have these QCLs associated to both colonization and response. So we can see if we can look within the plant genome and understand why, and boost the potential, therefore, of finding lines that really love the fungus and that could be good consistently in the field. So not only would we then have the right lines, but then we ask, well, are AM fungi even in rice cultivation? Are we promoting them in the way that we grow rice, and how can we boost that? So here we come again with IRRI, and Amelia's already introduced the direct seeded rice. So this involves directly sowing into the seed, into the soil, and it has much less water, especially in those dry aerobic systems. So this is what we're interested in, because we think the fungus is there because it's not waterlogged, so it's aerobic. And so we have these kind of breeding inputs that we can jump on the back of now as a mycorrhizal community and say, look, now we're in rice, but we're growing it aerobically. Now we can look for the fungus, and this is exciting. So we, a few years ago, we came into fields in the Philippines, and we just took one variety from the breeding program of Dr. Shalab Dixit, and we compared across different fields with different growing systems. So dry DSR that was either irrigated or only irrigated in like a rain-fed system, or we had DSR puddled or transplanted. And if we look in there, we can see that actually what we thought is true. We see colonization only in the case where there's no puddling and it's this dry aerobic system. So that's great. Now these systems are interesting. And we continue in collaboration with Amelia and Shalab, and in a lot of the, in some of the trials that Amelia was just talking about, we've come in and well, not me, the team in this room have come in and helped dig up the plants so that I can look at the roots and see, one, is this, all the work before was on traditional lines. Does this genetic variation that we see in those lines extend to that DSR breeding pool? And can we use that to our advantage? And also, how is the agronomy that is being promoted going to affect that? So we've taken samples, and that's why I'm here at IRRI, to analyze them from different varieties in different systems, and also in a nitrogen and irrigation trial. So we have different levels of fertilizer and water added, and I'm going to try and correlate the presence of the fungus with those different agronomies, the plant traits, that, especially the root traits that Amelia has already measured and has data for, and between the different cultivars. And so that brings me to the end. So to summarize, we're at the perfect time, especially as more and more people are thinking about water-saving aerobic rice, to come in and try to understand what this beneficial fungus could do in those systems. And with the right practice that promotes them, and also the right cultivars, now that we understand this huge variation between the different rice lines, we could be more and more looking towards how these could be used as a nature-based solution to reduce fertilizer inputs. And with that, I'll just acknowledge all the huge team that helped. So everyone in the Crop Science Centre, the Cereal Symbiosis Lab, everyone in Amelia's lab, and especially the field team who always helped me digging the roots from all those experiments, and Meg, and the team at WACI as well. So thank you.
Thank you very much, Emily. There's a question in the chat from Tim, but I'm going to take chair prerogative and ask a question first. I'm just wondering, what is the yield penalty between growing rice under a paddy system compared to an aerobic system? And do you think if there is a yield penalty, is this where greater opportunities exist with mycorrhizal fungi to offset that penalty?
I'm probably not the right person to answer the yield question, but as I understand, because a lot of these varieties were already bred for lowland rice, cultivation, then the yield that we see currently in DSR, especially if you put lowland varieties in that system, is lower. But that's why these breeding programs exist, to find, high yielding direct seeded rice. So whether mycorrhiza can help that yield, I don't know. And what I always scoot over in these talks is we don't really know what we're asking for in the mycorrhizal community, at least everyone who I've asked. Do we envision that we would inoculate with fungi? What fungi would that be? What mix, and what would they do? Or are we trying to promote native fungi? And that has quite important questions, because at the moment inoculum is really expensive and it's actually not very well regulated, so a lot of it is just dead spores and it's being sold very expensively as this great solution. So if you talk about yield penalty and economics, there's so many things to consider. I think at the moment the angle that I always come at is that fertilizer one, because that is easy to understand, and if we start there, then we can start asking how we see this is going to work. But yeah, whether we could get the same yield, I'm hoping that we can reduce the amount of applied fertilizer, somehow harness the fungus and get similar yields. If the yields always tend to be lower, then maybe we fall within these carbon credit schemes and we focus on people that are trying to grow sustainably in more like an organic farming situation. But I think it's something that we don't really talk about, especially my lab's mostly molecular. And when I do ask bigger names in the mycorrhizal community, everyone says something different. So sorry, that was long-winded, but that's my views on how we're going to get to with yields and what that actually means.
Thank you very much, and well dodged. Thank you. In a nice way. Tim, you have a question, so I'll pass over to Tim.
Yeah, no worries. Well, congratulations on doing so many mycorrhizal measures on so many genotypes. That's quite impressive. The question I have is, what's known about the interaction between parenchyma formation and AMF? I mean, does parenchyma make it easier or harder for AMF to inoculate a plant, for example?
So there's a body. I mean, rice has parenchyma, and we see quite high levels of colonization. Although I'm very biased towards our pot sand system in the glasshouse, so I don't think they inhibit it. But what a large body of research looks at is whether, because the fungus still survives in the water, it just has really low colonization. So it is there in paddies. It's not, there's a question in the literature of whether it's doing something. But a lot of people believe that it survives within the parenchyma because that creates an aerobic environment. And that's most of what I know about that.
Thanks.
Thank you. There's 3 other questions, and we'll try and answer them quickly if we could, just because I'm quite keen that we run to time. Paula, I'm not sure if you want to ask your question or whether you want to ask me to ask on your behalf. Yep, Paula.
Yeah, there was a comment earlier about how inocula is very expensive, and I was wondering what type binocular was used to do the field trials, or these single spores, or yeah.
So that was a species mix, which was locally produced. IRRI's next to the University of Philippines, Los Baños, and they have a technology hub that produced the inoculum because AUCA's quite keen to stay native.
Yes.
That comes with its own problems that then we're comparing a mix to the clean spores that we have in the lab. So, yeah.
Okay, thank you.
Adam, do you want to ask your question quickly?
Yeah, Emily, of course I know about the Blumenau method because I was your external examiner and I thought it was absolutely fantastic. But my question is, is that what you're using for colonization measurements in the field? And do you know that works when there are multiple species, or what you're doing?
We started in the field, but I have pushed back towards looking at the just like normal tripe and blue staining, mostly because I'm a bit worried about the genetic variation, which you talked about in the Viva, which is a small amount, but what about in elite? You know, we always would then have to test how much we're worried about genetic variation in Blumenau abundance. I also think it's quite quick to screen, especially when you're working in different countries, just with a normal light microscope. So Blumenau, you need shock freezing, which comes with its own problems in the field. So yeah, to be honest, I've moved back to staining, but I also have thousands of samples, so we'll see how long that lasts.
Thank you. And then one final question from Davide, who I feel I had to let ask the question because he had asked a similar question to Amelia. So Davide.
Yes, so thank you very much for your talk. I'm curious about the relationship that are not going according to plan, in the sense that upon application, plants, certain varieties are not responsive. So have you looked at the colonization of the roots? Are those varieties simply not engaging with AMF?
They're still colonized. So I don't really see a correlation between the extent of colonization and response. And I think that's important because we assume, I think a lot of people in the literature assume that it's more colonized, more beneficial, and it's not always the case.
Okay, thank you. Thank you very much.
Thank you very much, and thank you for the questions, and thank you very much, Emily, for a very interesting talk. So we're now going to move on to Helena Bochman, who's at the University of Jülich. I am hoping, there we are.
Yes.
Helena, do you want to share your screen?
Thanks for the introduction. I first would like to start with some words to Amelia. Amelia, congratulations for receiving such an honored award. I'm really happy that you got it, and also thank you very much for nominating me that I can present my work here. So hi also to everyone else. My name is Helena Bochman, and I would like to talk today about the role of rice root diameter undergrowth. Okay, let's start with who I am. So I saw myself listed in when I registered for this event as Dr. Helena Bochman. That is not the case yet. So my highest degree is a Master of Science, and I started working with rice in my master thesis, also together with Amelia, where I looked also at contrasting drought stress recovery strategies. When I finished my master thesis, I was really motivated to keep the rice thematic going on because I also thought that rice roots are really fascinating, and I was searching for a way of financing this. So Amelia recommended me the ACTSAF scholarship. ACTSAF is the Council for Tropical and Subtropical Agriculture Research. And right now I'm in my final year as a PhD student where I'm still looking at rice roots. Okay, I am not alone in this path, so here's some pictures of my supervisor panel. My doctor father is Uwe Rascher. My CGIR contact person, of course, is Amelia from IRRI, and I have two daily supervisors, Josephine Kant and Johannes Postma. In general, what I'm doing in my PhD is I'm screening a larger panel of rice lines, and I'm trying to understand how their root architecture and root morphology influence their performance under drought stress. And for that, I have multiple approaches. There were field experiments going on the Philippines, completely managed by Amelia and her team. Thank you already for that. And also, what I did here was modeling. I tried different phenotyping platforms, and I'm also using greenhouse experiments. And today, I would like to show you some results from the greenhouse studies. My basic motivation is that I would like to understand how root system architecture and morphology influence water uptake. So in general, here I like this picture. It's not rice, but I like that you can see so many parts are taking place in this water uptake. You have the shoot, the above ground part, you have soil, you have the rhizosphere, but of course also the root system architecture and morphology that I'm looking at. Okay, I first would like to present you the sensor I'm using. I'm using the SWAP original, and yeah, first published by Dr. Dagmar van Duschoten, and you can see here in the video how it works. So these rings are moving upwards as pots, and this is a time-lapse video, so normally it stops at every centimeter. And what it does, it can precisely measure soil water content in this specific depth non-destructively, and that over time. And to give you an idea how sensitive the sensor is, it's sensitive to the addition of around 20 microliter of water. As I mentioned already, if you really want to understand how the sensor works, I recommend you the first publication, but there were also another PhD student before me publishing papers, but he never worked with rice, so let's do it with rice. So first I would like to present you my growth conditions. So we had a 3-week establishment phase with well watered, which was around 20% volumetric water content in the greenhouse. And after that, I transferred my pots to the swap chamber that had roughly the same the same conditions, and I simply stopped watering the drought strands, whereas the well-watered plants were continuously watered. And then after 10 days of drought stress, I did a destructive harvest. So, for example, took the leaf area, and also here on top, you can see what I did with the soil core. I cut them in 5 parts, and I did root scanning for the architecture. So this has nothing to do with high throughput, but it's still interesting. Okay, so Amelia is always telling me don't forget the shoots, so I present you first the shoots of my rice lines. So that is Baramboro and Focati. On the left side, you can see Baramboro. On the right side, you can see Focati, and I would say that you can already distinguish them by eyes if you look at them, right? The shoot architecture is quite different. So Baramboro has the thin and longer leaves, whereas Focati has the shorter leaves, but they're wider. And also what I thought is that you can see the drought response in Baramboro quite intense, whereas it's not that easy in Focati. But if you compare, the total leaf area was really interesting, but within the same treatment, the genotypes were comparable. That means that, of course, you have a reduction of leaf area under drought, but if you, for example, compare the both control plants with each other, they had the comparable leaf area. And also if you look at the drought plants, they had a comparable leaf area. Okay, let's now move to the below ground part. There are 2 scanning pictures from the WinRiser system from the top soils. And I always think that if the above ground architecture somehow also represents the below ground architecture. So for example, on the left side on Baramboro, you can see that they had much more number of crown roots than Focati. On the other hand, of course, you have a reduction on the drought stress, but that is something you expect. And also, if you look at the diameter of this crown or nodal roots here, they are much thicker than Baramboro. So for the further slide, I will talk about the results, what we saw with these 2 contrasting lines, but I always confuse my supervisor with these names. So I decided to make it easier for you to rename these lines for the following talk to thin-rooted and thick-rooted. Okay. So let's start with a general water uptake plot. You can here now see 2 time points. That is early stress, that was right after I stopped watering the drought stress plants, and the late stress, that is close before harvest. And in the early stress, where of course you didn't have a treatment effect, that was simply because the drought stress plants still had water available in their pots, but you could already see here that the thin-rooted line takes up more water than the thick-rooted line. And if you look at the late stress, the thin-rooted, well-watered line took up the most water. However, the difference between the drought stress was quite immense for the thin-rooted line, that the drought stress took off less water in the end, whereas the thick-rooted line was managing to taking up comparable amount also in the last days compared to the well-watered plants. This is a plot you could do with a scale, right? So that is not what the, so the cool thing of this is that, oh, I'm sorry, I forgot to, I wanted to mention that you have here the both, these numbers are now the volumetric water content left in the pot. So I wanted to show you that they had a comparable difference between well-watered and drought stress treatment. Okay, but now let's also look at the depth profile. So you can see on the y-axis now the depth of the pot. 0 is the surface of the pot, whereas 45 is the really bottom of the pot. And on the x-axis, you can now see the root water uptake. So for example, for the thin-rooted, at the end of the late stress here, you can see that a lot of water is taken up here from the top, whereas the drought stress plant shifted this little uptake more to the bottom. On the other hand, the thick-rooted line had also, with the well water, took up more from the top. However, the differences between the thick and between the treatments were not so intense, and it's also more shifted for the drought stress toward the bottom. Okay. And the second that I would like to show you, that is also again a plot about a depth profile here. On the y-axis, you can see again the centimeters of pots, and on the x-axis now you see the soil water retention. So for everyone who's not normally working with drought, it's not only important to look at the absolute water content, it's also important because every soil has this individual capability of holding the water. So how strong is the water bound to the soil? And that is what the soil water retention explains you. So on the blue line that I added here as a help, that represents a 5% soil water content. The red one is a permanent wilting point. And on the left side, you can see the drought pots, on the right side control, and here we have thick-rooted and thin-rooted. So this orange line is day 26, so that is in the middle of drought. And you can already see what already showed before, that the thin root system takes up water faster because they were already drier here, right, than here. However, the really interesting part of this plot is at day 31. There is this dark brown line, is that you can see that regardless of their architecture, the thin and the thick-rooted line dries out the middle of the pot stronger than top and bottom. And that's the same here for the thin-rooted. And that is something you do not expect like this. You would expect something like a progressive drying with the top dry, and then it's get wetter to the bottom. Okay, so whenever I looked at this plot, one other thing that I thought was that the thick-rooted line always looked for me in the beginning here more homogeneous across depth, whereas the thin-rooted line had this really strong drying top and then this wetter bottom. So what I did was I compared 2 layers over time. So I always looked for every day at centimeter 20, so in the middle of the pot, and I compared it to the top 5 centimeters. And that is something you can now see on this plot. So on the x-axis, you have now days, okay? And the y-axis simply describes the difference between top and bottom. And the only thing that you really need to understand here is that as soon as a bar is positive, the middle was drier, whereas it was negative, the top was drier. So for example, for the thick-rooted at day 26, so the orange line here, if we compare the centimeters 20 to 5, you can see that the middle was slightly drier. Whereas if you look at the thin-rooted line, it strongly dries out the top first. When so it both ended up with a middle drier, but the pathway towards over the time was different. So the thick one dried out more homogeneous over the top, with a middle drier, and the thin-rooted with top drier. Okay, with that I'm already coming to my take-home messages. So here on the left, you can see again the thin-rooted, and here's the thick-rooted. So for the thin-rooted, I would like to remember you that the thin-rooted line takes up water faster. Both lines dry out the middle stronger than top and bottom. And the thick-rooted line takes up water more uniformly across depth and time. And I would be really happy in the end if you remember that for the answer, how do root system architecture, morphology, influence water uptake, that you remember that nodal root diameter matters. And with that, I would like to thank all the people helping me during my PhD with the Forschungszentrum Jülich, with the root group here on top, and my Dr. Father Uwe Rascher, of course. But I would also like, of course, to thank ATSA for financing me through the last years. And my last final thanks, of course, goes to Amelia, and I'm really happy that her team is sitting in the room. So I can also say thank you very much for helping me with all the fieldwork that happened at IRI, and thank you for your whole support. Thank you very much. Yeah,
Thank you very
a lot
much,
of people
Elena.
in my room.
Yeah, and yeah, you had 5 seconds left before the camera went on. So yeah, well done for keeping for time. No, it's fine. It's good. It's good.
Okay.
So anyone got any questions? Just put your hand up or put something in the chat. But the question I have is, so looking at the water distribution with depth, I don't think you showed any figures regarding root distribution down the soil profile. Have you correlated the two to see if the root size distribution down the soil profile correlates with these changes in water content?
Yes. So what I think I have it here.
The next one.
As an additional plot. Okay. So because that is a plot that I didn't want to show during my talk because it's so many bars and no one really gets it. But what you can see here on the y-axis is the soil depth. So because I cut it them in 5 pieces, right, for scanning. And the interesting part is that we have this the same or comparable root area. Of course, you have the most root area in the top, but if you compare in between Baranboro and Fulcati, it's comparable. So you have the comparable amount of root area with different architecture. Baranboro is doing that with a lot of thin roots, whereas Fulcati is doing that with less but thicker roots. And of course, you have less of what are you talking about maybe is also that if you look here at the middle, you have less root area, but it takes up more water. So yes, there is also a change in this water uptake per square centimeter of root area. Yeah.
Okay, and I presume you've got the same figure for root diameter then, do you? Based on what you were saying about root diameter driving root water uptake, I was wondering if you have any changes in root diameter with depth that might explain these potential changes in water uptake.
So the root diameter that I'm right now referring is a nodal root diameter, and this was and this was calculated for me over the whole depth. So I calculated the nodal root the average nodal root diameter over the whole depth. So yeah, you can really distinguish them by eyes. Yeah.
Okay, thank you very much. I'm going to go and invite Dennis if you'd like to unmute yourself and ask your question.
Yeah, sure.
Dennis Florentine, if you're not comfortable, I'll ask the question. So the question is, I'm curious why your setup includes 3 plants per chamber. Was this done to ensure lots of roots could be extracted at the end? I'm also curious how the chambers were watered. Was it from the top or from the bottom? And what was the rationale behind your decision?
Yes, that's a really good question. Thanks for asking it. So I decided in the end to put 3 plants in this pot, which is honestly, if you test the line, it would be too much. But I was really afraid because I saw that in my master thesis, this highly plastic lines, that if you give them quite a lot of time, they really change their root system architecture, right? And for me right now, I really wanted to see, okay, both lines, how they differ for well watered and drought stress. So I don't want it to have a quite plastic response. So I put in 3 plants to make sure that the drought stress was comparable fast, but the root system architecture would not change because in the end, otherwise I would have compared 4 different root system architectures and morphologies. So that is why I put 3 plants in the pot. And I'm sorry, I forgot the second part of the question.
The other one was how you actually irrigated the plants. So was it from the top or was it from the bottom?
No, we watered them from the top on a scale, of course, right? So you can always adjust them to this 20% volumetric water content in the well-watered condition. So we normally we always watered them from the top. The plants that got watered from the top. Yeah.
Okay. Thank you very much. Glyn, Glyn, I'm going to welcome you to unmute, or I can ask a question on your behalf. The choice is yours.
I can ask it. Thanks. Any ideas of the route distribution across the column, or would you get preferential growth of the routes down the outside of the tube? It's always a problem in many experiments. I'd like to hear what your thoughts. Thanks.
I'm sorry, can you go a little bit more? Could you repeat the question? So you're asking, so I
was wondering if the routes were growing preferentially down the outside of the tube.
Yes, some of them were growing. Yes, definitely. So that is, some of them were growing against the border of the tube. Yeah, yes. I didn't measure how many or how much percentage of that. So
Okay,
yeah.
Okay, thank you, thanks.
And there is a question from Adam actually that kind of like links in a little bit to this. Adam, do you want to ask a question?
Yeah, no, I like that. It's a nice design, this experiment. But my question is, I guess if you leave these plants long enough, there's enough roots to get every drop of water because the root length density is high enough everywhere. Do you know if that's the case, or because if it's the case, then we're surely looking at some other property of the roots, like the hydraulic conductance. It's about its preference for water rather than what water it can reach. So I don't know if that's a coherent question, but have you any thoughts on it?
That's a good question. I am. That's a good question. I will remind that for the.
Talk about that another time.
Yes, yes. I'm sorry. So that's a good question. I remember it, and I will put it in my discussion. Thanks.
And just one final question from, I'm thinking someone at the University of Aberdeen, but they're in proper camouflage mode and they're just down as UOA. But back to the root hair questions, or there's been a lot of discussion around root hairs. Would you consider looking at root hair length and density at the top, middle, and bottom of the root system?
Yeah. So I was thinking about it, but in the end we decided against it because of time reasons. Yeah. So we thought it would be an interesting question, but yeah.
As with everything, there's always lots of interesting questions that develop as you go along, but you never have the time to investigate everything.
Yeah.
So thank you very much, Elena. So we're now going to move on to the final presentation in this workshop, and that's Alexandra. So we're going to end this first workshop, and we're traveling to Senegal now for Alexandra, who works at the Institute of Research for Development in France, but currently in Senegal. So over to you, Alexandra. And as with everyone else, I'll put my camera on with 2 minutes to go. So if you can, keep an eye on that. Thank you very much.
Okay, thank you. So yeah, thank you very much for the opportunity. So today I'm going to talk not on rice, but on pearl millet and on some work we've been doing with our colleagues here in Senegal at the Senegalese Institute for Agricultural Research, but also with colleagues from the University of Nottingham. So as an introduction, so pearl millet is a crop that has been domesticated around 4,500 years ago in West Africa, and it's very well adapted to Saharan conditions, where it when it grows in low input agroecosystems, it will yield something when other crops like maize, rice, or wheat will tend to fail. And so therefore, it's a very important crop for food security in West Africa. And it's also a cheap and very high source of proteins, but also micronutrients and vitamins for the population in these regions. Yet, pearl millet yield remained low, and its yield is much affected by drought, by climate change. And as you can see in this map here, the impact of climate change is is much stronger in the Sahelian regions where pearl millet is grown. And as a matter of fact, a study, a modeling study comparing modeling pearl millet yield to actual pearl millet yield here in black already showed that climate change, and especially variability in high heat and high rainfall events, have already decreased in average 10 to 20% the yield of pearl millet in the past 50 years. So it's important to increase or to improve pearl millet still to drought stress. And what we've done in the team is really to sort of characterize a little bit better the drought stress patterns in West Africa. And what we did was to collect the climatic data in Senegal, where in the region where pearl millet is grown, for the past 20 years. And we fed that data into a crop model that estimated shoot biomass and yield. And after some clustering analysis, we observed basically 3 patterns: no stress in green, a vegetative or early drought stress in blue, and the terminal drought stress in red. But what was quite interesting, in fact, was that we observed that in the past 20 years, the drought stress occurred more often than the terminal drought stress, and actually it had, based on the simulation, higher impact, a stronger impact on grain yield and shoot biomass as compared to the terminal drought stress. So we decided to focus on that specific drought stress for our studies, and we asked the question, what could be the contribution of root traits in mitigating the impacts of vegetative drought in pearl millet? And to answer this question, we used a pearl millet genetic association panel. So it's a diverse panel of pearl millet coming from all the way across Saharan Africa, but also India. And response in the field in Senegal, and in the same plots, in the same fields, we also harvested roots on a very specific root on node 4 for root anatomy, and we studied their root anatomy using laser ablation tomography in collaboration with our colleagues at the University of Nottingham, and we tried to find links between the anatomy of the roots and the drought response. So as I mentioned, we performed these experiments in the field, so in Senegal, at the station of Bambé, during 2 years. So we had 2 years of trials during the dry season of 2021 and 2022. We used 160 fully sequenced pearl millet in bread lines, and we had 2 treatments. The first one was the irrigated treatment, where the plants were irrigated all the way through until maturity. And the drought stress treatment actually started after 21 days after sowing. We let the plant establish a little bit, and then we stopped irrigation for 3 weeks. At 42 days after sowing, we harvested the plants for root phenotyping, and then we re-irrigated the field from that point on until maturity, where we measured agromorphological traits such as biomass and yield and so on. And in total here in this experiment, we had 4 replications. And of course, we followed the dry down in the field using volumetric water content. And we saw that in the drought stress treatment, so this is just an example, but we saw that for both of the experiments, that the water content reduced during the drought stress and go below the onset of stress, which is considered as this red line here. And then it increased again after we re-watered the field. So first, we looked at the drought stress response in terms of grain yield and morphological traits to sort of have an idea of the diversity in drought response in this panel. And as you can see here, the drought stress had a very strong impact on shoot biomass in both years. So it was reduced by around 60%. At maturity, the shoot biomass recovered somehow, but still we had a significant impact. But in fact, it was quite surprising, or I mean, in a way, comforting that the grain yield was reduced in the drought stress treatment as compared to the irrigated treatment in the same range as the predicted values that we obtain using modeling, so around 40% in the vegetative drought stress treatment. So we tried also to better characterize the stress tolerance by measuring a stress tolerance index that we measured on grain yield and also on shoot biomass after the drought stress, at the end of the drought stress, but also at maturity. And for that, we use this index here, which we thought was interesting because it not only it gave us an idea of the drought tolerance, but also an idea of the plant performance under irrigated conditions. And that's very important for breeders. So we thought this index would be nice to use. And for this index in the panel, we saw a nice diversity, showing that nice diversity in drought stress response, and also a good correlation between both years, showing that our results were quite robust. And what was also quite interesting is that this drought tolerance index for grain yield actually correlated positively and significantly with the drought stress tolerance index for shoot biomass at the end of the drought stress. So in other words, plants that were able to maintain their shoot biomass at the end of the drought stress were also able to maintain grain yield and high yield under drought. So that was in terms of root anatomy. So as I mentioned, we used a laser ablation tomography in collaboration with our colleagues at the University of Nottingham. So we could image all the roots. And in fact, in pearl millet, we had a root scan version of a pearl millet version of root scan that worked quite well. And we were able to segment all these traits. Yet we did not use all of them. Some of them were more robust and reliable. And these are here. And as you can see here, we tried to correlate these traits with the stress tolerance index, but also with the grain yield under drought. So these traits were measured under drought. And what was quite striking here is that in both years, we saw a positive and significant correlation between the maintenance of grain yield under drought and the grain yield under drought with total metaxylem, the mean area of metaxylem. So that was a very strong result for us, very robust. And in fact, if you consider the literature, you may think that this is quite surprising in a way, because it has been described more or less the reverse in the literature. So anyways, we decided to follow up on that and try to better understand how these metaxylems could contribute to drought tolerance. And in fact, they obviously contribute to axial water content in the roots to supply the shoots with water. So we asked the question whether they had an impact on plant transpiration. So we ran an experiment in the greenhouse using the exact same experimental design that we used in the field. The pots were placed on the top of scales in the greenhouse, and we had a panel of contrasting lines. So we had around 20 lines that were contrasting for total metaxylem area. And we did this experiment in two types of soil. A soil with 50% sand, 50% potting mixture, which is basically peat, and a soil with 100% potting mixture, so peat. And for this experiment, so at the end of the dry down, we measured plant morphology, so leaf area, root biomass. We also measured the root anatomy, of course, and we measured the transpiration response demand. So you can see here, so some plants might respond very strongly to the evaporative demand, and other plants less. We would show less response to the evaporative demand. So when we performed this experiment, we then looked at the transpiration response, but we decided to focus not this time only on total metaxylem surface area, but try to find a more physiological, I would say, parameters. And we thought of measuring actually axial hydraulic conductance, which is very much correlated with total metaxylem area, and that is the sum of individual axial hydraulic conductance of all the metaxylems here. And we thought it would represent a little bit better, it would account better for the number also of metaxylem rather than just the total surface area. And so we classified our genotypes within two groups with high axial hydraulic conductance and small axial hydraulic conductance. And of course, we made sure that these two groups also showed similar shoot biomass. because we wanted to see specifically the effect of root axial hydraulic conductance. So when we looked at the transpiration response at the last day of the experiment, we saw in the irrigated treatment in both soils that the plants with large metaxylem area transpired more, as you can see here in the blue line, when the evaporative demand increased in both soil, and plants with smaller metaxylem areas, it transpired less. So some of these points are and I would say that's sort of expected. And in fact, this translated into more water used in plants with larger metaxylem area or larger axial hydraulic conductance as compared to plants with smaller xylem hydraulic conductance. Look at the drought response this time. We saw more or less the increase along the day. So they used more water, you can see here. But in fact, very interestingly, in the drought stress treatment in sandy soil, we observe the exact opposite results in a way that plants with larger metaxylem actually transpired less than plants with smaller metaxylem vessels. And that was also significant for some of the points here. So if you translate this transpiration here into transpiration rate by normalizing by measuring the water loss per second per centimeter square of leaf, you can also calculate a slope here that will give you actually to the evaporative demand. It would be an indication of the transpiration response to the evaporative demand. So when we measure this slope in both of the groups of genotypes, we again confirm the result completely opposite, where you can see here that in the pit soil, plants that, sorry, the phone is ringing, but it's not for me. Plants with higher axial hydraulic conductance show the positive correlation with the transpiration rates, meaning that plants with higher hydraulic conductance were more responsive to the evaporative demand, while in the drought stress in the sandy soil, plants with higher axial hydraulic conductance were less responsive to the evaporative demand, so their slope were lower, and therefore they were restricting their transpiration in response to the evaporative demand. So we tried to summarize that into a working model that we have at the moment, where we consider that in sandy soil, in wet sandy soil, when the evaporative demand, in the roots, so plants with a smaller metaxylem vessel, as the demand from the shoot will increase during the day, will more faster be limited in their water supply. So therefore, they will show less water use and more transpiration restrictions. Now, when you stress experiment, similarly, when the evaporative demand, this time we consider that the limitation will be within the soil, and especially because in sandy soil, it has been shown that the hydraulic conductance will drop much faster than in other soil, and definitely more faster than in pit soil. So our hypothesis is that for genotypes with higher axial hydraulic conductance, we would reach, when the evaporative demand would increase, a faster drop in the root to soil, sorry, soil to root axial, soil to root hydraulic conductivity, because the plants will suck water much faster, and therefore this gradient in the rhizosphere will lead into a transpiration restriction when the evaporative demand will be high. And this might be linked to improved TE, then transpiration efficiency, and of course we save water within the soil and would be linked to the drought stress terrains. So in conclusions, what we think, what we've done is to actually apply field phenotyping for anatomy on pearl millet and the drought in the field in Senegal. We've shown that metaxylem vessel size correlated positively with pearl millet performance under irrigated and drought stress conditions, and that was actually linked to transpiration restrictions. So larger metaxylem were showing increased transpiration restriction at high evaporative demand, and that was dependent on the soil that we used. So that was quite interesting because here we have a trait that can link soil hydraulics and plant transpiration in the soil-plant hydraulic continuum. So next, what we aim to do now is to find genetic loci associated with metaxylem features that we are employing GWAS at the moment to do that. And also we have the question of metaxylem plasticity, because we know that this trait is plastic. It's also plastic dependent on and this plasticity dependent on the genotype. So we have a high G/E effect here. So we also want to identify the genetic determinant on of that plasticity. And as you can see here, I'd like to acknowledge a lot of people. So these are contributed to the root harvest in the field. So that was huge work. So all the people at IRD, the people, our collaborators in Serras in Senegal, colleagues from Andrea Carminati, Mathieu Javot on the soil side, and all the colleagues from Nottingham, our founders also, of course. And if I have just one more minute, maybe, I'd like to say thank you to Amelia. Of course, I mean, Amelia, you've given me my first opportunity as a postdoc. I was working in the lab, you brought me to the field in India, in Nepal, everywhere. And I learned a great deal. I mean, basically, I learned everything that I know. And in a way, you disseminated your passion and your enthusiasm to me because I'm still working in the field today and still working on roots. So thanks a lot, Amelia, and many congratulations. And all of the speakers said, thanks also a lot to your amazing group in the Philippines at IRRI headquarters. That was the best time of my life, actually. Thank you so much.
Thank you very much for a very interesting talk. Now, I'm going to selfishly say we have time for another couple of questions, but unfortunately I've got to go do my own presentation somewhere else in 5 minutes time. So we'll restrict the questions a little bit and try and run to time. Tim, straight in there with a question. Tim, do you want to ask
Alexander, fascinating project, really. It's really interesting. I just wondered if there was any noticeable differences in the shoot and anatomy. So, for example, stomatal size, number, density, etc., between the small and the large metaxylem varieties.
Yeah, looked at that a little bit. Actually, Dylan Jones, a PhD student that was working with Darren Wells at the University of Nottingham, looked at that. I'm not sure if he found consistent results in terms of shoot anatomy and the link with the root anatomy. I don't think it was really the case. We haven't looked at the stomatal density or stuff like that.
Okay, thanks.
Thank you. If anyone's got any questions, just raise your hand. I do have a small question here. I interpreted this wrong. Were you reporting metaxylem volume per each plant, or was it cross-sectional area? And I was wondering what the relationship was between root length and transpiration.
Yeah, sorry, it was cross-section metaxylem area. It was not volume. Sorry for the confusion there. And in terms of link with root architecture, actually, we also tried to look at that. So we did not see any differences in any impact or any linked between metaxylem area and root biomass. So that's one. We looked at the branching density also, lateral root density between the two groups of genotypes. Well, we have preliminary results showing that there's no differences in terms of branching, but that would need some confirmation because we've done that only on small portion of the root and also on root from node 4. So we will definitely need to confirm that, but definitely this is an interesting question. And in terms of integrated phenotypes, as Amelia mentioned, for sure metaxylem might be linked to other phenotypes, and whether the synergy between architecture and anatomy there, we will investigate that in the future.
Okay, thank you. Adam has a question as well. Adam, do you want to ask the question directly?
No, it wasn't a question. It was a comment.
Oh, sorry.
I just think it is beautiful. It's a beautiful experiment in demonstrating the complexity of plant-water relations, the importance of roots, and most importantly, the importance of soils. Yeah, it's really nice. And I think the soils often get overlooked.
Thank you. Thank you.
And there was a question from Dennis who's just clarified he isn't able to ask a question directly because his mic doesn't work. So he was wondering if you think there is a way to integrate soil type based on PSA maybe into your model. Thanks in advance.
Yeah, actually, in this so for the first modeling aspect that I presented, we use SSM. So that one, I don't think there's a soil component in it, but we've developed actually OpenSimRoot for Pearl Millet, and OpenSimRoot has a soil component. So we've used the older version of OpenSimRoot, not the new version where you can simulate drought stress. So we are trying to implement that at the moment, but in a bigger scale. We are also trying to work on connecting FSPMs with crop models to sort of better understand the role of root anatomical traits, depending on the soil texture, but also on the climatic, evaporative demands.
Okay, thank you. And then one final quick question from Leah, which is just a question about why you're using the fourth nodal route rather than other ones. Did you do any pre-work on assessing which route to be using for this particular study?
Yes, done a pilot experiment actually on 20 genotypes contrasting to determine which route we wanted to use. So basically, we used route number 4 because it was the easiest to collect. That was not the most important reason. The most important reason was because it actually grew around the time of drought stress imposition. So when we are around 21 days, when we started our drought stress, this root started to come out. So it was one of the root growing during the drought stress. And the last reason was also because roots from node 1 to 3 were quite thin, are quite thin in Pearl Millet, and we had issues to LAT them, to image them with LAT. So that's basically the 3 reasons.
Okay, thank you very much. And I'd just like to say thank you to all the speakers that spoke in this particular workshop. So I will do a virtual round of applause, but thank you very much, everyone. I'm now going to pass over to Tim, actually, if you want to just clarify the timetable.
What happens next, yeah. So yeah, thanks for the speakers in the last session. It's really interesting. We're going to have a break now for lunch in the UK, or at least in UK, Europe. So we'll stop for 30 minutes, well, actually 25 minutes now. So we'll be back at 1:20 UK time, where we have 3 more speakers who will continue our journey through the rice root and rhizosphere, and then 1 speaker who will talk about barley root angle at the end. So please come back after a short break for lunch in 25 minutes or so, and we'll start again. So have a nice break.
Thanks very much, Tim. Hopefully we've got Jasmeet online. I'll do the same as the previous chairs of the sessions. I'll turn my camera back on a couple of minutes before the end of the allotted time, and then we'll take questions from the posted in the chat. So to introduce Jasmeet Kaur Bahambra. She previously did her PhD in Aberdeen and is now working at the University of Copenhagen in Denmark. And she's going to talk to us about rice biological nitrification. So Jasmeet, if you share your presentation.
Hi, so I'm Jasmeet and I'm currently working in Copenhagen, but for the last 5 years, I was in Aberdeen. And today my talk will be slightly different. So it's going to focus more on the microbiological aspect of rhizosphere. So my talk today will be focusing on nitrification in rice. So nitrification is the process where ammonia is converted into nitrate. And the key microbes involved in this process are ammonia oxidizing archaea and ammonia oxidizing bacteria, so AOA and AOB, which mediate the first and rate limiting step in this process. The end product of nitrification, which is nitrite, can cause groundwater pollution, and nitrification can lead to nitrous oxide emission, which is a potent greenhouse gas. but in natural settings, the interplay between plant nutrient uptake and nitrification keeps the ammonium and nitrate in balance, which does not cause these environmental issues. In agricultural settings, however, the fertilizers are applied to increase nitrogen usage efficiency of plants. Rice is one of the highest has one of the highest fertilizer application rate of around 120 kilogram per hectare per year, and 60% of it is lost due to nitrification process, even in paddy fields. While application rate of fertilizer has increased in rice agriculture yearly, it does not correlate well to the nitrogen usage efficiency increase in plant systems. This only increases the load on agricultural economy and increases contribution to greenhouse gas emissions. And for this, I would be mostly focusing on nitrous oxide emission, which contributes to around 3 to 5% of total emissions from the total nitrous oxide emission. So 3 to 5% is from rice agricultural systems, and the emission rates increase if rice are grown with intermittent flooding growth. And to combat the increased nitrification in agriculture and to increase the nitrogen usage efficiency of agricultural systems, we can identify plant species that can suppress soil nitrification through rhizodeposition. So many grasses, including rice, have been known to possess this ability where they can inhibit ammonia oxidizers in soil through certain root exudates. And this process is called biological nitrification inhibition, or BNI. And BNI is a relatively newer field, so not a lot of it is yet known. So the aim of my project was to combine plant physiology and microbiology to understand the factors influencing BNI efficiency of rice in soil. So the aim of this project was to shed light on the following topics: the impact and spread of BNI compounds within the different soil compartments using ammonia oxidizer abundances, to understand how the BNI compounds inhibit the two different groups of microbes, that is ammonia oxidizers, to correlate BNI efficiency to different plant traits, and to establish a testing method that can correlate BNI efficiency detected in soil to lab settings. So for the experiment, I chose 5 different rice genotypes with variable BNI efficiency, and these 5 genotypes were previously also detected to have BNI activity based on a hydroponic setup where the root exudates were tested against a single model ammonia oxidizer culture. So in my experiment, what I did was I grew the plants in soil microcosm, where I used a mesh bag to separate the rhizosphere soil compartment with the extended rhizosphere soil compartment, and the bulk soil was a separate pot, which was unplanted pot, and the plants were sowed in the mesh bag or the rhizosphere compartment. And the plants were grown for 90 days till flowering stage, and they were sampled every 30 days for different analysis. So the first part that I would like to look at is the inhibition of different groups of ammonia oxidizers in soil compartment. So plants release BNI compounds as both hydrophilic and hydrophobic components in their exudation. However, during the release of these, the highest concentration of the exudates would be in the rhizosphere, and they would spread out slowly into the rhizosphere, and only the hydrophilic portion would be spreading out, and the hydrophobic portion would be near the roots and would remain in the rhizosphere. So what we expect would be the ammonia oxidizer abundances would decrease the most in the rhizosphere soil compartment. To analyze this, we did a plot where we had the ratio of abundance of ammonia oxidizers, where the ratio was determined by the abundance of ammonia oxidizers in the rhizosphere or rhizosphere extended, where the plants were planted in, against the bulk soil abundances where there was no plant in the soil. And as such, anything higher than this one would mean that the growth of ammonia oxidizers was stimulated in the rhizosphere or rhizosphere extended compartment, and anything lower than one would mean that the growth was inhibited in the rhizosphere or rhizosphere extended compartment. So coming to day 0, for all the genotypes, I've combined the results for all of the plant genotypes and both ammonia oxidizer groups together. And for day 0, the abundances in all the 3 different soil compartments was same because the ratio is 1. At day 30, we already see an inhibition in the rhizosphere, where the ammonia oxidizer abundances were lower than 1, but no such effect was seen in the rhizosphere extended compartment. And then this trend was the same at day 60 and then day 90. So what this suggests is that the BNI compounds within the rice that are produced are mostly concentrated in the rhizosphere, which would also suggest they are possibly hydrophobic in nature. Next, we wanted to see the inhibition of the different ammonia oxidizers in soil. I did a previous study where I looked at pure BNI compounds, and what we observed was that the archaea were more inhibited than the bacteria by pure BNI compounds. So what we expected in this study as well is that in the rhizosphere, ammonia oxidizing archaea would be more inhibited than the bacteria over time. So this is the detailed graph of what I showed you earlier, where the graph has been broken down with respect to genotype and the different ammonia oxidizer group that I was testing. So, but if we only focus on the ratio of abundances in the rhizosphere, I did a linear modeling of the decrease in the ratio of abundances over time for both archaea and bacteria. So when we do a linear modeling decrease, what we see is the death rate of ammonia oxidizers in the rhizosphere, which is what is represented here for each different genotype. And what we can see is that the BNI efficiency is variable with respect to the different rice genotypes as well as between the 2 ammonia oxidizer groups. And if we look closely, archaea are more inhibited in the first 2, that is the high BNI genotypes. And in the last group, last plant genotype, that is the low BNI genotype, this suggests that although the inhibitory effect on ammonia oxidizers is variable and is a genotypic effect of rice, archaea are disproportionately more inhibited by BNI compounds. So this is again the same plot, but where I've combined the death rate or the BNI inhibition efficiency of both archaea and bacteria together, where I got one plant that had really high BNI efficiency, which was Lemont, and then 3 plants with kind of similar-ish BNI efficiency, and Azucena with the lowest BNI efficiency. Next, we tried to correlate BNI efficiency to different plant traits. So previous studies with forbs and grasses have shown that root exudation rate negatively correlates with root morphology. And in this study, I thought it will be nice to see a relation as well on a more finer scale between different genotypes of the same plant. Because in this study, they used different plant species. So in addition, we also hypothesized that the BNI efficiency of plants will be positively correlated to root exudation rate. So basically, higher the root exudation rate, higher would be the BNI efficiency. So we collected and dried root exudates of 90-day-old plants. Here we did a linear model to establish a relationship between the root exudate weight on the y-axis and which was collected after, which was dried root exudates collected after 24 hours of exudate collection, and the dry root weight of the plant. And what we see is a really nice negative correlation between root exudate weight and root dry weight. I also managed to look at the root length of the primary root of these plants, and the exudate weight had a similar negative correlation with the primary root length of the plant. And when we look at the root exudation rate relationship with BNI efficiency or death rate of ammonia oxidizers in soil, we see a positive correlation as we expected. So this suggests that the smaller root structure produces more root exudates, which in turn would increase the BNI efficiency in soil. Lastly, we wanted to establish a better assay for in vitro detection of BNI activity using cultures. So in vitro detection of BNI in exudates is performed by collecting the root exudates and then testing its inhibition against a model ammonia oxidizing bacterial strain. On the other hand, detection of BNI inhibition in soil depends on looking at changes in the absolute or relative abundances of ammonia oxidizers in soil. So several previous studies with different plants, including maize, sorghum, have shown, and also rice, have shown that the in vitro detection inhibition using the model ammonia oxidizer strain and root exudates collected, so the BNI inhibition mediated by root exudate collected and tested on model organism strain and using the soil method does not correlate with each other. So to demonstrate this in this study, I compared the BNI efficiency of the 5 different strains I selected and its BNI efficiency in soil to what was the inhibition that was observed in the previous study, which was used to select these 5 genotypes. And you can see that the trend of inhibition is completely different between the 2 different studies, which is a huge thing if we want to find out a unified way of understanding BNI efficiency of plants. So in our study, what we did was, instead of testing the BNI efficiency in cultures on just one single strain, we used multiple species of ammonia oxidizers. And on the graph on the y-axis, you see the inhibition percentage of ammonia, percentage of the growth of ammonia oxidizers in presence of root exudates and of the different rice genotypes. So here the archaeal strains are represented in red and the bacterial strains are represented in blue. And as you can see, the variability in inhibition is quite varied between the different strains and also between the different rice genotypes, which was obviously expected for us. So if I then do a modeling of the response of all 6 strains combined together, we get a trend of high to low inhibition. And when we compare it to the inhibition in soil, it's not very different to each other. In fact, the trends are very similar. So in both cases. So this suggests that a multi-strain approach is better to get a microbial response, which I think is the same in the case of non-ammonia oxidizing microbial responses as well. And hence we have SynCom communities to test this. So it would be nice to have a SynCom community developed for this system as well, but it's hard. So to sum up, we identified different rice genotypes with differential BNI efficiencies, which was stable both in soil and culture assays. We found rice plants release the BNI compounds very close to the rhizosphere, suggesting the presence of more hydrophobic BNI component in rice exudation, and that the BNI activity in plants can be linked to plant physiology, where a decreasing root mass and increasing root exudation rate increases the BNI activity of the plant. So the work I presented here was done in my PhD at University of Aberdeen, where Cecil was my supervisor and Adam was a huge help in designing and conceptualization of the project. Thank you for letting me present here.
Thanks very much, Jasmeet. We have a couple of questions in the chat. So, Tim, would you like to unmute and ask your question?
Sure. Nice talk, Jasmeet. Really interesting. It was really interesting to see such a strong correlation between exudation rate and BNI capability, and particularly Lamont being very good at what it did. I just wondered if you had any idea about what makes up those root exudates. Is there any obvious candidates for what's causing the BNI activity in those exudates particularly?
So I didn't present it here. We did identify a new BNI compound, which is hydrophobic from these root exudates. And there have been a few more hydrophobic BNI compounds that have been isolated from rice root exudates. I haven't presented the results here, but we also looked at 16S diversity. So I've not completely analyzed the results, but it seems that overall Lamont root exudates produce general allelopathic compounds as well, because the diversity was very low.
Cool. Okay, thanks for that.
Thank you very much. Korat, would you like to unmute? Perhaps not. So I'll ask the question on their behalf. They've asked why root exudates were only collected for 90 days when you perform other experiments on different time frames.
I did collect root exudates for every time frame. It's just I was at the end of my PhD, and day 90 promised the highest BNI efficiency. So I analyzed the root exudates from day 90.
Okay, thanks very much. And then there's apparently Korat doesn't have a microphone. So Dennis, would you like to unmute?
Dennis also doesn't have a mic. So
Dennis also doesn't have a mic. So they say, hi, Jasmeet, a nice presentation. I was wondering if you could compare specific root exudates released in the rhizosphere when using hydroponics versus when using soil growing methods. I assume that distinct root exudate classes will be released depending on the medium and substrate.
Yes, so it's not a project that I'm doing, but a project that I am a collaborator with, and it's happening currently in Cecil's lab, where Cecil's postdoc is looking at differences in BNI efficiency between hydroponic growth setup and soil growth setup. So, but it's happening in wheat, so maybe the results would be out by next year, possibly.
So there's room for work in rice as well.
Yeah.
Great, and thank you very much. In the interest of keeping to time, I'd like to move on to the next speaker. So thanks very much, Jasmine.
Thank you.
Then our next speaker is Chris Tembo from the University of Aberdeen, and he's going to be talking about his PhD work. So Chris, can you share your slides?
Yeah, thank you. I'm Chris, and I'm actually going to present part of my research work as a PhD student at the University of Aberdeen. My studies were funded by the Commonwealth Scholarship Commission in the UK. So, yeah, that's the outline of my presentation. As we might be able to know, rice is an important food crop for over maybe half of the world's population, and it actually provides a lot of global calorie intake. But the crop is predominantly grown under paddy conditions. So because of that, we have a lot of methane emission from the crop, which results into high carbon footprint. Actually, it's one of the most important producers of methane. So because of that, we actually need to find ways maybe to, because we are fighting global climate change, as such, we need to reduce the carbon footprint of the crop. This is one way maybe can be done through use of aerobic conditions, or maybe growing the crop in conditions that do fluctuate between aerobic and anaerobic condition. And we do believe that if we are using such systems, then what we call avascular mycorrhizal fungi would become so important in the crop, as they seem to have a lot of benefits theoretically, as well as what has been proven scientifically in the field and in the greenhouse conditions. So what are these avascular mycorrhizal fungi? Actually, Emily has given a good introduction to this. They do belong to the Glomeromycotina subphylum of the Mucoromycota. They are also quite ubiquitous, which means they can be found in almost every environment out there. And they are, because they are in the soil, they form symbiotic relationships with plant roots, especially the grasses, which are important, and rice being an important food crop, they all seem to have quite they make a huge association with them, and studies have shown some benefits in terms of that. So this is a representative of the association that might be formed between a plant root and a fungus. In this case, we have this blue line, which shows the hyphae going into the root cortex, forming very fine structures, which we normally call the avascos, as we can see here. And these are structures which are used for exchange of nutrients. So as mentioned earlier on, the importance of this AMF or avascular mycorrhizal fungi is that they are quite important in terms of nutrient uptake, as they can be able to get nutrients from far places, or maybe in areas where the plant roots will not be able to go through. So mostly it's going to be inorganic phosphorus and nitrogen, but also in terms of water uptake and protection from different stresses, including drought, as well as salts. So why are we focusing on this? As I said, my study is focusing on the biological significance of the genetic variation in rice root colonization by avascular mycorrhizal fungi. So this stems from a research study that was done by Davidson, who was also part of the lab that Adam chairs. So they carried out a study where they used a rice diversity panel 1. So So what they did was to grow rice in stellar soil conditions. And then during that study, they established that there is quite a huge variation in terms of colonization rates. So some of the rice cultivars had a lower percentage in terms of colonization from 22 to some which had 90%. But of course, the majority of them were in the middle in terms of colonization rates. So this gap, we know it's a wide gap, but we don't know what's the benefit of these crops in the middle. I mean, these crops which have high colonization, what do they benefit from the mycorrhizal association? So there is that biological mixtures gap that we need to understand. So based on that, we had some research questions. So 3 of those research questions are the ones that we are going to address in this presentation. So the first one was, how does rice root colonization differ when inoculated with different AMF species? And how do different rice cultivars respond if we give them the different species of AMF? And the last one is whether the response after being inoculated is proportional to the rate in which they get colonized. So the first one, we used a pot experiment in the greenhouse. So I'm going to talk about 2 experiments. So the first one is a pot experiment where we grew 5 rice cultivars in 5 AMF treatments. So we had Rhizophagus irregularis, Pneumophis mosii, Claroidium, Cladogrammus claroidium, and a mix of these 3 plus a non-mycorrhizal treatment. So this is the setup that we had, and actually it's after 4 weeks of growth. So we measured plant height, we assessed the number of leaves, number of tillers, and at the end of experiment, we weighed the plant roots as well as shoots. We also analyzed the phosphorus content, that's total phosphorus and total nitrogen. And finally, we also measured the colonization rate for each of the cultivars that had been inoculated. So the results that we have, the first talking about the rate of colonization that we had. So we can see that in the experiment, different rice cultivars had different colonization rates. So some, if we look at the first one for Azucena, for example, Azucena had a higher colonization when it was inoculated with Pneumophis mosii, but it had a lower colonization when it was inoculated with Cladogrammus claroidium. But the most important result would be because we had a significant interaction tail, so we can see that the colonization was actually dependent on which cultivar and which AMF species was used. So we can think about one of the varieties here, the first one. We have BJ1. BJ1, when it was inoculated with Pneumophis mosii, it had a higher colonization up to over 90%. But then when it was inoculated with Claroidium, the colonization rate decreased to somewhere around 10%, which is a huge difference. But if we also consider Chuan 4, for example, Chuan 4 had a very low colonization of around 14% when it was inoculated with a mix of the species. That is, when we mixed Claroidium, we mixed Pneumophis and Rhizophagus irregularis. But then, if we consider the same Claroidium, the colonization with BJ1 was much higher. So that only tells us some story about it, which we can talk about maybe competition because of the mixture of the species. So in general, the Claroidium consistently was a low colonizer. The same observations were also made for a number of abascos and a number of vescos in the when we try to assess it. The second question is, how do different rice cultivars respond to inoculation with different AMF species? So in this case, we are looking at growth parameters such as shoot dry weight, plant height, number of leaves. But for the sake of this presentation, I'm only presenting on shoot dry weight, but the results are almost the same. The pattern is just the same. So for all the plant growth parameters, cultivars and AMF treatment, they were significantly different. But in general, mycorrhizal plants were actually very low, which means they might have been affected somehow. And if we consider, for example, if we look at this graph, which looks at the plant shoot dry weight, we see that this, looking at Azucena, when inoculated with Rhizophagus irregularis, shoot dry weight was quite high, which means it was a very tiny plant in comparison to the non-mycorrhizal treatment, as well as if we compare with the Claroidium. This is the Claroidium, as well as the Phenoliformis, because these generally had the same consistent level in terms of colonization. That's the Phenoliformis mossiae and Rhizophagus irregularis. But you can see that they are quite different in terms of the shoot dry weight. In general, Bala as a variety, or as a cultivar, was not generally affected by any of this. So this is Bala, and that's how the general shoot dry weight that it had. In terms of nutrient uptake, if we consider phosphorus, phosphorus was not affected in terms of uptake. But when we looked at nitrogen, nitrogen uptake was generally affected. And in one of the crops, however, it was not affected. For example, Bala. Bala was, in terms of the amount of nitrogen that it took, it was almost the same as the non-mycorrhizal treatment. Next, we try to look at the responsiveness of each of the cultivars when inoculated. So generally, there was some kind of negative mycorrhizal growth response, especially when these crops were subjected or were inoculated with either Phenoliformis mossiae, Rhizophagus irregularis, or a mix of them. But to some extent, we have seen that this Claroidium actually had some kind of a positive effect. The third question was, is the response of rice to inoculation proportional to the rate of colonization? According to our results, it doesn't seem to be the case. If we look at this chlorogram, we see that what we have, high Phe content, abascos and vescos, they all have some kind of no correlation with the with any of the plant growth parameters. So the last part of the research is where we took the same understanding to the field, where we did the field study in Malawi in conjunction with Lilongwe University of Agriculture. So we used a commercial inoculant, which contained Glomus interradices, Glomus mossiae, Glomus aggregatum, and Glomus etnatum. That's according to the suppliers. And we grew 10 rice varieties, which we believe are upland tolerant varieties. So colonization was different across when we included a commercial inoculant, then we had some kind of significant difference in terms of how colonization happened. And this colonization were also improved with time. So the first one is for hyphae at 42 and hyphae at 91 days. That's the second graph. So you can see the difference. However, there was no effect in terms of any of the yield parameters. So this is the graph. So I will not talk much about it, but there's no effect. So the most important part is we observed variation in terms of colonization at different time points. So what we try to understand is some of these varieties, they seem to be consistent. For example, this Wachango here, it is consistent in terms of colonization. At 42, it is somewhere around 30-something%, and at 91, it is around 40%. So it is relatively low in terms of colonization. But then we have this other one, which is seem to be acting the same, but at least in terms of when you look at 42, it is one of the higher colonizers. And then we have this one, which was somewhere around Kayanjamalo, which was somewhere around 35%, but then it increased to somewhere close to 81. So these were becoming interesting varieties. So what we did was to try to understand if there is anything that is happening within the crop, within the plants itself. So we know that there is going to be multiple co-occupancy by the mycorrhiza in the field. So is it linked? Is this what we observed here? Is it linked to the changes in the AMF fungal composition? So what we did was to do sequencing of the DNA for these 4 cultivars. And then the reason is to establish the composition and abundance of the species. Is it affected by the cultivars, or maybe at different time points, is it changing? So that has been done, and we are waiting to see the results. So in conclusion, there is a wide genetic variation in rice root colonization by AMF fungi that has been there, and F. mosii and Rhizophagus irregularis, they seem to be higher colonizers as compared to Claroidium chromas. But there's also growth retardation, which is especially true for Phneriformis mosii and Rhizophagus irregularis. However, we cannot clearly link colonization rates to the plant growth or maybe nutrient uptake. That's the end of my presentation.
Tracy, you're muted. You're still muted, Tracy.
Just thought I'd try and undid that. Yeah, thanks very much, Chris. We've got time for a couple of questions. So David had a comment on one of your slides. So David, do you want to unmute and just explain what you were looking at?
Yeah, it's one of the graphs where you showed us the various cultivar with the type of inoculant and their percentage of colonization. So I struck with the fact that the mix had pretty much the same pattern of F. mosii alone, although let's say shifted towards a reduced colonization. I just wonder whether you have any idea or suggestion as to let's say what are the cause for that.
I'm not sure if I got the question correctly. Is it for?
I think it was for the slide where you had the colonization rates for the different single AMF and the mix across the 5 genotypes? Was that the one, David?
So it looked like a roller coaster. That's it.
Yeah.
This one. So looking at the green and the red, they basically overlap. They seem just shifted. I just wondered, what are the ideas that are circulating in the lab to explain this phenomenon?
Yeah.
My understanding that the mix contains FMOSI, right?
Yes. So maybe that would be some kind of an explanation, but we don't know because it's supposed to contain FMOSI, chloroidium, and so possibly we might be thinking that the FMOSI is kind of dominating, but I'm not so sure about that. So that's just my speculation.
No, I was curious. I mean, if that's okay. That's just it got my attention. This very similar pattern.
Yeah. That's true.
Thanks very much, David. And Tim?
Thanks, Tracy. No, I was really interested in how wide the range was of mycorrhizal inoculation from virtually 0 up to 90%. And I just wondered if there's a correlation between the strength of mycorrhizal association and the root hair phenotype as well. So, for example, Azacina has a very high mycorrhizal inoculation. Does it have a very short root hair, for example?
Thank you for that question. I would struggle to answer that question because I have not really studied if I mean the different root structures for these ones. So they were not part of the study that I did. So yeah.
Okay.
Not to worry. Might be interesting to look at.
Yes, I think yes.
Cool.
So thanks very much, Chris. I think in the interest of time, we better move on. There are a couple of other questions in the chat. So it would be nice, Chris, if you were able to answer those too. Next speaker, we're going to swap over to Bali. And it's my pleasure to introduce Gwen, who now works in Tim and my lab. She did her PhD in Germany and then worked in Saudi Arabia and joined us approximately a year ago. So over to you, Gwen.
Thank you very much, Tracy. Obligatory question, can you see my slide?
We can.
Great. Cool. Thank you, the organization of the workshop, to invite me. And it's actually a great pleasure because I've attended this workshop 2 years ago remotely. And then last year when I came here, it was basically the first thing that I did, and I attended in person. And now giving a presentation here is a great pleasure for me. I'm going to talk about the genetic control of the barley root angle today. So we will of course stick to roots, but go a bit more to the molecular part of it. If I manage to move my slides, yeah, great. So I don't, because this is a root-related workshop, I don't have to all convince you about the importance of roots and how important they are for plant health and yield. But I would like to draw your attention how important it is in which layers the roots are actually located in the soil. If we look at the soil profile, the nutrients and resources of the soil are actually located in different layers in agricultural soils. So for example, if we look at immobile nutrients such as phosphate, they are more located at the topsoil. And I'm trying to get a pointer here. Great. So immobile nutrients such as phosphate are more located at the topsoil, for example, while mobile nutrients such as nitrate are located at the topsoil, but then also leach through to deeper soil layers. And similar thing goes for water. So if we have rainfall, more water is located at the topsoil first. And then once the soil starts drying, we get more water in the deeper soil layers. So it's actually very important where exactly the plant places their roots in the system. So for example, we would assume that roots that are located at the topsoil are able to better access those immobile nutrients that are located there. While if we have roots in the deeper soil layers, they might be able to better pick up water that is there after a drought or mobile nutrients such as nitrate. And how these roots are actually placed in different soil layers is determined by the root angle, which is set by what is called the gravitropic set point angle of roots. So this angle describes the angle of a plant organ in relation to gravity, which depends on the root type or the plant organ type, the environment like the soil, and also developmental stage of the root. And in my project here at the Hutton, I look at the barley seminal root system for that. The barley seminal root system is an amazing model for that, for the gravitropic set point angle, because the seminal root system consists of a lot of seminal roots that have approximately the same developmental stage and age, as you can see here on the picture of a like 5-ish day old barley seedling. So they all have the same genetic background, same age, same developmental stage, but they all have different gravitropic set point angles. And what is very interesting about the set point angle is that it's not only set once in the life of a plant, but it can be maintained over a specific developmental stage. And here in such a classical rotation experiment, you see that the roots have some kind of an intrinsic memory of which angle they are set to. So if we look at the lower one here, this angle, this root is growing in a roughly 45 degrees angle before the rotation, and after rotation, it's still roughly 45 degrees. So it just keeps continuing to grow in this angle. While this guy here is roughly 90 degrees before the rotation, and then adapts its growth direction very fast back to roughly 90 degrees. The gravitropic set point angle is, as I said, somehow related to gravity. So what happens in such a rotation experiment is that the roots basically sense the new center of gravity in the root tip, where there are starch-filled granules that perceive the gravity because they are more dense than the cytoplasm and therefore sink down, and thereby trigger a signaling cascade that involves a relocation of the auxin distribution, but also changes of pH and calcium signaling. And then the signal is transduced to this cell elongation zone, where cells now differentially elongate. So in this case, in this example, the upper cells elongate more so that the root growth is in the end directed downwards again. And we know already a lot about this signaling mechanism, but we don't know a lot about how this graviperception is linked to the formation of the angle and how the plants maintain such a specific angle per root over their lifespan. In previous work, we have already identified a very important factor in barley that controls the root gravitropism that we call enhanced gravitropism 2. It was isolated from a barley mutant population. And on the left, you see the wild type, which has several seminal roots with different gravitropic set point angles, while in the mutant that we found, all the roots are growing basically 90 degrees downwards. And this holds true also for later stages of development, where you can see here that in the wild type, the ground roots are also spread in an angle, and the roots, both the crown roots, but also the lateral roots in the mutant grows deep down in our mutant. We wanted to find out what is actually wrong in this mutant, and therefore we did such a classical rotation experiment that I've shown you before. And you can see in the upper lane that the wild type slowly adapts back to the new direction of gravity, but the mutant is actually much faster in the direction. So we therefore assume it shows a hypergravitropic, a hyper reaction to gravity. So that's why we call it an enhanced gravitropism. We then tried to figure out which mutation is actually causing the phenotype, and used whole genome sequencing and with a combination of byte segregant analysis to narrow it down to a specific candidate gene, which encodes for a sterile alpha domain containing protein. And our mutant actually has a mutation that encodes a stop codon in front of the functional domain. To understand if we hit the right candidate, we made CRISPR lines and were able to remove quite a bit fragment of exon 1 from the gene. And indeed, if we look at the phenotype here, again, we can see such a steep root phenotype. So we were quite convinced that we found the right candidate. We then wanted to understand if this is also true for other plant species, and looked at a mutant of both rice. Lots of people have been talking about rice, so it just came to my mind. No, we actually looked at a mutant in wheat, where both homologs of EGT2 were mutated, and also here we see a steeper root phenotype. So we assume that this gene is actually the gene function is very conserved. To understand what EGT2 is doing and where it's acting, we looked first at its expression, and with an RNA in vitro hybridization, we found that it is expressed in all the tissues that are relevant for gravity signaling. So the root cap, the meristem, and then also the elongation zone here. To understand what EGT2 is doing, we compared the transcriptome of wild type and EGT2 plants, and specifically we looked at roots that are already growing in like the vertical angle to not have any bias between the root angles in our analysis. And we focused on the root cap, the meristematic zone, and the elongation zone in S tissues, because those are the ones that we know are relevant for gravity signaling. And interestingly, we found a lot of differentially expressed genes, but the most striking ones were in the elongation zone, because there were a lot of genes that are related to cell wall processes. By now, we have already somewhat analyzed the cell wall composition and didn't find a major difference, but I would also like to look more at the mechanical properties of the roots, because this is only transcriptomic data, and we don't actually know how that translates to the mechanical properties yet. However, in parallel, we also had a look at a different hypergravitropic mutant, which was called EGT1, not because the gene is related, but because it was isolated from the same mutant population. And here we found that the cell wall stiffness of the cortex actually differs between wild type and EGT1 mutants. So our working hypothesis at the moment is that the cell wall stiffness in the cortex is somehow regulating basically the reaction to, or the response to gravity, and kind of in the wild type, it kind of makes a counterforce against the gravity so that roots can grow in an angle. And if you mutate EGT1 or maybe also EGT2, the roots don't have this counterforce anymore, and that's why they grow steep down. So in my future research, I would want to look more in the role of EGT2 for the cell wall composition and for the general properties of the roots, and also look more at the EGT2 signaling network and how it's actually acting. I've told you before about the root gravitopic set point angle. So I said that roots are somehow able to remember the set point angle over time. And to now find out what makes the roots different and how they can actually remember this different angle, I'm using another, again, a transcriptomic approach to look at the transcriptome of roots with different angles. So I've chosen a vertical one, and then a medium one, basically, and one that grows very shallow. And I now compare the transcriptome of these three root types. And again, I focus on the zones that are important for gravity perception. So the root cap, the meristem, and the elongation zone, which I isolate by laser capture microdissection. And I hope to find gene expression that is correlated with the root angles, and that then might somehow explain how those roots are set to different angles and how they can maintain them even after a rotation. And interestingly, so this is only preliminary results, but interestingly, I found most of the gene expression changes in the root cap. So I did not actually find a lot, which was not very surprising because the root samples I took coming from the same plant, so they have the same genetic background, grown in the same conditions, same age, same everything, except for the angle, that's the difference. So I didn't expect a lot of changes. But interestingly, in the root cap, I found a gene expression that is actually correlated with the angle. So here, the one on the left is the transcriptome of vertical roots, then the medium roots and more horizontal roots. And from these genes that are where the expression is somehow correlated with the angle, I would, in the future, in the next year, like to isolate some candidate genes and then analyze them in more detail. For example, look at mutants, look into a haplotype analysis to find if the genes have a function in root angle regulation. And I've talked a lot about the genetics of the root angle right now, but why might that be interesting for agriculture? So the idea would be that if we have root systems with different angles, like for example here on the left, root system with a very steep angle might grow deeper down into the soil and are therefore maybe better for drying conditions, while we have shallow root systems here on the right, which might be better for low phosphate soils, for example, because they can forage for phosphate on the surface. And so far, there hasn't been a lot of examples where we find out that that's actually true. I want to show you here one example from actually rice. So going back to rice here, where there's one nearly isogenic line that has a steeper root growth in comparison to its wild type. And in a drought stress experiment, the group in Japan could show that these plants with the steeper and therefore deeper roots are actually more resistant to drought. And in another experiment here, I would like to use, or I'm using at the moment together with my honor student, the wild type barley together with the EGT2 barley with a steep root system in a rhizotron experiment. And we want to compare their performance under drought and want to find out if such a placement of roots in different soil layers is actually beneficial for drying conditions. And if we understand better how EGT2 works and also how root angle is regulated, and then if we understand which meaning that actually has for plant performance, we can hopefully in the future adapt our crop plants better to difficult soil conditions. With that, I would like to thank everyone who is involved in the EGT2 story, my current collaboration partners, and everyone at the Hutton who's contributing here. I might have missed some names, and my funding. Thank you very much.
Thank you very much, Gwen. So, as previous, if you'd like to ask Gwen a question, just drop it into the chat. As usual, Tim has one. Tim, would you like to go ahead?
Yeah, sorry, I keep asking questions. So I just wondered what impact selecting for root gravitropism might have on the ability of the plant to cope with changing stress in a season. So plasticity, I suppose, is the term. But you know, imagine a situation where the plant's dealing with low phosphorus and then it becomes drought, which is the bigger problem. So locking in gravitropism, will that have an impact?
Yeah, I mean, the honest answer is we don't know yet because there are very limited studies about the root angle, the role of the root angle in general. And then mostly only under one stress and not combined stress over time, but for sure using EGT2 with this very steep root system is not going to be the solution for everything. It's probably only the solution for a specific environment, probably a well-fertilized environment. And I guess for environments where we cannot really predict the conditions, we probably would be more interested in having a root angle that is more plastic and can maybe be better adapted at that time for the specific conditions. But so far, yeah, I would say everything is this research is still at the start, so we have to kind of look at one condition at a time at this point.
Sure, absolutely. Thanks.
Adam?
Yeah, nice that you. I mean, it's nice work, and it's of course very interesting. And you mentioned DRO1, so I recently realised something really odd about DRO1, and that it was identified in an IR64, which is our famous shallow-rooted check. But if you look at the allelic variation in that gene, IR64 has a loss of function mutation, which is unbelievably rare in rice. I think there's only about four rice varieties in 4,000 that have that allele. So my question is really, what about natural variation in root angle? Because I'm sure there is as much in barley as there is in rice, and whether there is the studies of that tell you whether or not there is value in breeding for deep rooting. So where do the deep rooting ones come from? Is that connected to low water availability or high phosphorus availability? In barley, as in rice, can we actually get some handle on relate traits around root angles, root depths to actually where natural allelic variation comes from?
There are some studies in barley about root angle variation, but to my knowledge, they have not really been mapped to any specific environment. So it seemed, I don't think that is known. I can mostly talk about EGT2, and there's basically no natural variation. So there's only one haplotype, only the full functional version of EGT2 and all the haplotypes that I could get hold of. So it's, and for EGT1, this other hypergravitropic mutant, there is some variation, and they also have a variation in root angle. So it seems like that this very steep root angle, like we shown for EGT2, where every root is growing down, is probably not very beneficial naturally. So there is no natural variation here. If you knock it out, the plant is probably not very happy under difficult conditions.
Thanks very much. If we've got time just for a couple more questions, I've got one from Kuat again. Do gravitropic effects in roots also influence when they interact with soil structure and soil stiffness and nutrient availability?
The fair answer is we don't really know yet. I mean, we only see the effect of the reaction to gravity in the root angle, and those studies haven't, there's not a lot of studies about it. So we don't actually know what the effect is under lots of different stresses at the same time.
Thanks. And I think it will probably have to be the last one from Leah.
Hi, thank you. It was a great talk. I found it extremely interesting. I found it particularly interesting how the central sort of vertical taproot was the one that had the transcriptomal sort of uniqueness. So in that chart you showed, it was the one that was lit up in red. And I was wondering what your hypothesis was for why that was the case. And I maybe thought that it might just be because at least one root at all times needs to get anchorage and get deep. So maybe the central one is the one that's most strongly encoded to do that. Would be perhaps my hypothesis.
Yeah, I mean, in barley there's no actually, or in cereals in general, I don't know. In barley there is no such thing as a taproot. So all the roots that you are seeing are the same root type. So the anatomy is the same, and they only differ in the angle. But yeah, you would always have at least one or two roots that grows deep down, and I guess this is just because it's important for anchorage. And then you wouldn't want, as a plant, I guess the other roots grow in the same angle so that you can cover more soil space.
Very interesting. Thank you so much.
Okay, thanks very much, Gwen. Can I just thank all the speakers from this session and everybody else, and then I'll just pass back to Tim.
Great. Well, thank you, Tracy, and thanks for that session, all the speakers. It was really, really interesting again. Yeah, I'd just like to thank all the speakers for today and congratulate Amelia on receiving the Dundee Root Medal for this year. As I said, we've recorded today's session, so we'll be putting it up on YouTube in the coming weeks. So when we do, I'll send a link around. But thanks for joining today and thanks for sticking around all day to listen to the talks. I think it's been a really interesting day. So thanks, and we'll see you again next year for the 2025 Dundee Root Medal. Thanks.